So good morning, everyone. Welcome to the fourth Phenotype Prediction Using Genomic Data workshop. As a reminder, this workshop was initially idealized by a couple of grad students at the time, Patricia Munoz and Marcia Resende. And the general objective of this workshop was, first of all, to bring to one room research scientists that were working on phenotype prediction, both in plant, animal, and in humans. But also to have an opportunity to stream it live so that people all over the world could actually follow research that was being done by top scientists in the area. So I'd like to begin, first of all, by thanking our sponsors for this event. First of all, the University of Florida Agronomy Department, as well as the School of Forest Resources and Conservation, have all contributed to this, to sponsoring the event. The UF Genetics Institute, UF IFAS, and finally also Rapid Genomics. I also like to thank three people that have greatly contributed to this. First of all, Allison, Cornelia, and also Joseph Kelly, who have done a lot of the work, administrative work, really to make everything work regarding registrations, regarding streaming, regarding the website, and so on. Without further ado, I'd like to pass the mic to Dr. Pat Concannon, who's going to give a few words from the Genetics Institute.
Okay, I'm all wired up here. So it's really a pleasure to once again have the opportunity to introduce this workshop. And I have to say that now that it's gotten to be the fourth annual, I'm beginning to lose track. That may be a function of my failing memory or just the success of this program. We're going to have to switch over to Phenotype Prediction 2016 or something because I won't be able to keep track anymore. I really enjoy this workshop and its connection with the Genetics Institute because phenotype prediction is one of those multidisciplinary, really cross-cutting issues that we all have to wrestle with. So, I mean, yesterday afternoon, when I actually had a few minutes to do some science, I was wrestling with a bunch of data having to do with variants of unknown significance. So, you know, variants in a single gene, high risk of breast cancer, hard to predict whether a particular variant is deleterious. I've got all kinds of software programs I can use to do that, but those software programs don't agree in their predictions. And if I take any one program and feed it new sequences to build a new conservation matrix, I can get different predictions out of the same program for the same position. Okay, and that's just dealing with a single patient with a single rare variant in a single gene. You know, when you scale up to multifactorial diseases where suddenly we have all kinds of different inputs we can use for prediction, we've got even added problems of complexity. And I would argue that while thinking medically, you know, medically is one way to think about how this impacts humans, perhaps the bigger public health issue is what are we going to do to clothe, house, feed the population of this planet. And once you start thinking about those issues, then you have to think about prediction in species that may have very long times before traits manifest. So trees for lumber, for building, cattle for food. And that's what makes this workshop so much fun, is that we take this basic problem and spread it across all these different disciplines and different applications with a host of really good speakers and an engaged audience both locally and internationally. So welcome to everybody, and I'm looking forward to a really great session. Thanks very much.
Our first speaker today is Dr. Francisco Penegaritano. If you have trouble saying his last name like I do, just call him Pancho. And he's going to be talking about a number of items related to phenotype prediction, one of them the introduction and the use of different levels of genomic information to support prediction models. So, thank you.
Thanks, Matthias, for the introduction. So thanks to the organizers for the invitation. It's really great to be here. Let me see if I know how to use this. Yeah, there you go. So this is the outline of my presentation. I'm planning to present about three closely related topics. I will talk about gene set analysis as what we call a post-GWAS tool. And then I will talk about how we can incorporate biological information, including results from this gene set analysis, into prediction models. And in the last part of my presentation, I will try to convince you that even if we do not have gene set information or pathway, we can actually infer de novo networks using multiple sources of biological information. So for this audience, it's obvious that genome-wide association studies have been very successful tools. Now, you will agree with me that GWAS studies focus on genetic markers with the strongest evidence of association because we have to deal with this multiple testing thing. And these significant markers explain, in general, only a small component of the genetic variance, and hence they provide only a limited understanding of the trait under study. On the other hand, what we call gene set analysis are becoming very popular tools. So basically, gene set analysis is a strategy where we try to test the association of a set of functionally related genes. So in a gene set analysis, multiple contributing factors are considered simultaneously, and hence, rather than focus in only one significant marker or one significant gene, we try to group and test a set of genes. And hence, this approach, we believe, provides unique opportunities to identify genetic mechanisms and pathways underlying complex traits that we, in general, we are interested in livestock, plant, and humans. So a couple of years ago, we described in detail how to go from a GWAS to a gene set analysis. And basically, this approach has three major steps. The first one is what we call the assignment of a SNP to genes. Well, in a GWAS, we work with the SNPs. In a gene set analysis, we work with genes. So we have to do the link. And the link is quite easy if we say, okay, I will assume that a SNP can flag a gene if the SNP is within the gene or is near the gene. In this case, we use 15 kb either upstream or downstream the gene. The idea to use 15 kb is to capture regulatory regions that we know are outside the gene, like the promoter, but near the gene. The second step is now to assign the genes to what we call functional terms, functional categories, or what we call pathways. The definition is quite simple. We believe that a gene set is just a group of genes that share some particular properties. For example, they are involved in the same biological processes. They have the same molecular functions. And for that, we have been exploring different pathways, different databases. We started working with gene ontology. We use KEGG pathways. We explore Interpro, that is a relatively new database of families of proteins. And recently, we are exploring what we call the medical subject headings or MESH. So the last step is actually the association analysis between a functional term and a phenotype of interest. And for that, we just perform what we call a Fisher-Isaak test. The idea here is to search for pathways that show an overrepresentation of significant genes. So this is a classical 2x2 table where there are three key parameters here. S is the total number of significant genes in my study. N is the total number of genes that I could evaluate in my study. And M is the total number of genes in a given pathway that I'm investigating. And the p-value of observing has significant genes in the pathway is given actually by this formula that comes from a hypergeometric test. So in the next few minutes, let me show you two studies in dairy cattle genomics where we apply this idea of gene set analysis. I will start with a paper that we just published 1 month ago about leukosis incidence in dairy cattle. This is really cool. Bovine leukosis virus is actually an oncogenic virus that infects B cells. The importance of the bovine leukosis virus is that it's closely related in terms of the genome organization and also the progression of the disease with the human T-cell leukemia virus. Now, as you can imagine, bovine leukosis has a really negative impact in dairy cattle production. And because no treatment or vaccines are available right now, a potential genetic solution, it seems really important in our field. So this is why we performed this study. Briefly, I want to describe the data. We use leukosis data from roughly 12,000 Holstein daughters of 3,000 sires. We have genotype data from 1,000 of those sires, and we perform a classical multi-trait GBLAP model. Multi-trait because we include milk production and we include somatic cell score in our models, and we use a single-step approach. Here we have the results. Classical Manhattan plot. We have in the x-axis, we have the chromosomes. In the y-axis, we have the percentage of genetic variance explained by 20 SNP windows. Here, the response variable is leukosis yes, leukosis no. So when you get this result, you say, no way, nothing. As you can see here, we couldn't detect any genomic region in the bovine genome that explain more than 0.4% of the genetic variance. No major genes, no peaks, a classical complex trait possibly modulated by several genes of small effects. The good news is we use the result of this GWAS, and we use our pipeline and we perform a gene set analysis, and look at this. We identify terms closely related with leukosis and retrovirus infection. The first one, the most significant KET pathway, is actually a pathway related with leukemia. That is a really, really cool result, because actually leukosis in cattle resembles the leukemia in humans. And actually we have now a list of significant genes that we want to explore. Another really significant term was this G protein couple receptor signaling pathway. It turns out that actually leukosis virus use these receptors to infect the cells. And another significant term was this regulation of nucleotide metabolic process. We started to read, and there are some data that have shown that the metabolism of the nucleotides are really impacted in some type of leukemia. So we started from a GWAS, no major genes, no candidate genes, no peaks. We incorporate biology, and we think now we have some results that provide insight into the genetic architecture of these complex traits in dairy cattle. And this result opened the door for future studies on resequencing and fine mapping of genes, and of course opened the door for the development of novel strategies for dairy cattle breeding. Briefly, I will discuss the second study. That is a manuscript that we have under review right now. It's about bull fertility, also in dairy cattle. As you can imagine, fertility is an extremely important trait in dairy cattle. So far, most studies have focused on cow fertility, in female fertility, while bull fertility, male fertility, has received less attention. However, significant percentage of reproductive failure in dairy cattle is attributable to bull subfertility, and this is why we think it's important to try to unravel the genetic basis of bull fertility in dairy cattle, and this is why we perform this study. So here we use almost 45,000 records of bull fertility that we have in almost 11,000 Holstein bulls. Almost 8,000 of these bulls had genotype data, we use different genome-wide association mapping approaches, including single-step GBLAB and a classical GWAS for accounting for population structure. But what I want to show you, this is another Manhattan plot. We have in the x-axis, we have the chromosomes. Here we have the genetic variance. You will agree with me that here we have some peaks, fortunately, and we can identify some candidate genes. Now, no region explains more than 1.5% of the genetic variance, and this is why, again, we pursue this idea to, okay, let's do a gene set analysis. Let's incorporate biological information and use the result of this GWAS. And look at this. We identify several terms that are closely related with sperm physiology and male fertility. You will see GO terms like fertilization, reproductive process, mesh terms like spermatozoa, sperm capacitation, sperm motility. We are working with sire conception rate. So it's really, really cool to get this result when we just go to the GWAS and we incorporate biology in that and get these pathways. We got this snare binding and SNAP receptor activities as a really major significant term. It seems these proteins are involved in the acrosome reaction. And we got also many terms related with ion transport and channel regulation, mainly related with calcium. And calcium is well known as a regulator of different sperm biology steps, including the acrosome reaction. So again, this integrative approach, we call integrative approach because we use the GWAS, we bring biology, and then we try to enrich the result of the GWAS study, reveals not only in this case genetic variants, but also molecular mechanisms underlying a really complex phenotype like bull fertility in dairy cattle. And this result can provide opportunities for improving bull fertility using genomic data. Doesn't want to work. There you go. So the question here is, beyond using gene set analysis as a post-GWAS tool, beyond using gene set analysis as a tool to better understand the biology behind a trait, to enrich the result, can we use this valuable information for prediction? More in general, how can we incorporate biological information into prediction models? Well, there are different approaches that we can use. One potential approach is to use different subset of SNPs to construct different G matrices, different additive relationship matrices. When I'm talking about different subset of SNPs, for example, I'm thinking, why don't we use what we call the genic SNPs? Those are SNPs that are within genes or really near genes. Again, this 15 kb probably depends on the species that you are working. We pick up this 15 kb in order to capture regulatory regions. Another potential subset is this, what we call the gene set SNPs. Okay, let's use SNPs that are actually within or near genes that belong to relevant GO mesh terms. So this is a way to incorporate the gene set analysis now in prediction. Why don't we use a subset that we call significant SNPs? Someone is calling? So maybe it's my mom, say that I'm in the middle of a talk. So another thing is what we call the significant SNPs. So the significant SNPs are SNPs that are marginally associated with the phenotype of interest. So imagine you can run a test and sort the SNP in order of marginal association and use those SNPs to construct different G matrices. So this is an ongoing project that we are working with Rostam Abdullahi. Rostam is here in the audience. We are trying to predict bull fertility using genomic data and biological information. We use sire conception rate as a measure of bull fertility. We have almost 8,000 bulls with both phenotypic and genotypic data. We use what we call alternative G-BLAB models because we construct alternative G matrices and then we fit different G-BLAB models. In fact, we fit different kernel models and we use the additive kernel as the G matrix. And then we evaluate the predictive ability of our models in cross-validation. That means we evaluate the predictive ability of our model in the testing set. We use a 5-fold cross-validation and we replicate this 5-fold cross-validation 5 times. So in terms of the different subsets that we are exploring, we have what we call all the SNPs, 54,000 SNPs after quality control. These are the SNPs that we can use spanning the whole genome. Then we have what we call genic SNPs. These are the SNPs within gene or actually near genes. We have 26,000 SNPs in the bovine genome using this chip that are what we call near genes. Third, we have the repro SNPs. This is funny. Repro SNPs are the SNPs that are within genes that are in the GO term reproduction. Why reproduction? Because reproduction was really significant in our previous study. And the fourth subset is what we call significant SNPs, 13,000. We run what we call a really dirty GWAS, not controlling for population structure, and we test for marginal association between the phenotype of interest and each SNP at a time. We sort the SNP and we use we use in total 13,000 SNP for that. Here we have the results. In the y-axis we have the correlation or the predictability of these G-BLAB models in cross-validation. As you can see here, when we use all the SNP together, G-BLAB, we get a correlation of roughly 0.34. When we use only the genic SNPs, the first result that we get exactly the same correlation that using all the SNP together. This is nice so far. Then for the repro, we expect that we will get a lower predictability because we are using only 1,000 SNPs. Anyway, the correlation is not too bad. And when we use the top significant SNPs, this is a cool result. We are using 13,000 SNP, and actually this model outperformed the model using all the SNP together. Now the question here is, okay, you are showing that with 26,000 SNPs that you call genic SNPs, you get the same correlation as using all the SNP. Show me the result when you take 26,000 SNPs, but at random. So try to test a null model where you use exactly the same size of the G matrix, but taking a SNP at random. Okay, there you go. When you take 26,000 SNP at random, actually you get the same predictability that using the 26,000 SNP that you call genic. So actually, it's not the genic SNP who is driven the predictability in this case. For the repro, the same. So the repro subset is 1,000 SNPs. We take 1,000 SNPs at random and we replicate this 5 times in each cross-validation and we get exactly the same result. Now, for the top SNPs, you can see that the predictive ability that we achieve with the top SNP, with 13,000 SNP, if we take at random 13,000 SNP and we replicate this, we do not get the same predictability. So in this case, we can say that actually this improvement in prediction is driven by this pre-filtering that we are using incorporating some biological information that in this case is just simple as a marginal association between one marker and the phenotype. So the second approach that we are evaluating right now is the use of multiple kernels. So when we use one subset of a SNP to construct a G matrix, we are using basically a single kernel approach. And the different GBLAB models, as you can see here, they fit actually different number of SNPs. So the next step is to use actually multiple kernels. It's a more flexible model. The model per se fits always exactly the same number of SNP, the total number of SNPs. But what we do here, right now we are exploring two kernels, but we can explore more, is actually we split the SNPs in two kernels and try to capture try to explore how we can predict better using two different subsets of SNPs in this case. So we will have again the predictive ability in the y-axis. In blue, we have the genic models. So as you can see here, a kernel model using one kernel with genic and a multiple kernel using a genic kernel and a non-genic kernel, what we call the polygenic, okay? This is important. All these models fit all the SNP that we have. So this model fit two kernels, one with the genic, one with the non-genic. We don't get any improvement in predictability. For repro, as you expect, when you move from one kernel with only 1,000 SNP to a model with two kernels, 1,000 SNP for the repro genes and 53,000 SNPs as the polygenic, now you get the same predictability as a model with one kernel using all the G-SNPs. And this result is really interesting. A model with one kernel using only the top SNPs perform as well as a model using two kernels, one with significant SNPs and one with non-significant SNPs. So this is an ongoing project. What we are doing now is we are exploring, actually, we are trying to disentangle this genic and non-genic and incorporating maybe exon and introns. And we are here actually incorporating information from mesh terms, trying to explore other mesh terms and incorporate that in the prediction models. But as I will say later, we believe that the multi-kernel is actually a natural approach to try to introduce biological information in our models. So the question now in the last part of my talk is, instead of using predefined functional terms, instead of go and say, okay, these are the geo-term reproduction that some molecular biologists discovered some years ago, is it possible to infer de novo networks? In other words, how can we integrate multiple sources of biological information and reconstruct networks de novo? I will try to convince you that this is possible. The main challenge here is actually try to decipher the flow of biological information from the genotypes to the endpoint phenotypes. In other words, the challenge here is try to integrate multiple sources of biological information in order to try to unravel what we call causal networks that underlie complex phenotypes. The question is why you want to deal with causal inference. We know causality is really tricky. It's really controversial. So why you want to deal with causality? Well, you will agree with me that actually decipher causal relationships between variables is probably the ultimate goal in most studies involving complex traits, by several reasons. The first reason is because if we know the causal relationships among variables, we have a better understanding of the biology of the trait. But for us today, if we know the causal relationship among variables in a system, we can use that information in order to predict the behavior of complex systems. So the idea when we deal with these multiple sources of biological information and we try to infer the networks underlying, is we work with a set of correlated variables. So for this audience, it's obvious that correlation does not imply causation. This is what we know and we learn in Stat 101. Now, sometimes we forget that if two variables are truly correlated, then there are potentially three underlying processes explaining that correlation. One is actually that X has an effect on Y, or could be that Y has an effect on X, or could be actually that there is no link between X and Y, but there is a third variable, typically a confounder, that actually has an effect simultaneously in both variables. And this is why these two variables are correlated. So the question here is, how can we go from data that we observe? And here I have 4 variables. It could be genotypes, gene expression, phenotypes. This could be DNA methylation. This could be proteins. This could be metabolites. How can we go from observed data and try to infer actually the network, but the causal network, beyond correlation, the causal network underlying this data? Well, for doing that, we have to assume that the pattern of conditional independencies that we observe in the joint probability distribution of this data is compatible with the causal structure. Let me explain this concept. Let's assume that we know the true causal structure. Simple model, 4 variables, x1 and x2 that has an effect on y, and y that has an effect on w. This is the true model, and let's generate data from this model, okay? We generate data from this model, and then we go and see what are the pattern of conditional independencies that I expect to observe in this data that was generated by this model. Well, we will expect, and we have to observe actually, that x1 and x2 are marginally independent. That means the covariance between x1 and x2 is equal to 0, and the correlation too. We have to observe that x1 and y, x1 and w are actually marginally dependent. That means the covariance between x1 and w is different from 0. The same from x2 and w, the same from x2 and y, and of course we expect an association different from 0 between y and w. Here comes the beauty of this approach. When we start to condition, things start to change. Now, if we condition on y, we expect now that x1 and x2, that they were marginally independent, now are marginally dependent. And if we condition on y, actually we block the path from x1 and w, so we expect that if we condition on y, now x1 and w are conditionally independent. So it seems it's easy to go from left to right. It seems it's easy that if we have the true causal structure, we can actually read out, we can interpret the conditional independencies that we expect to observe in the data. Now, if we know the process and we calculate all the conditional independencies that we have in the data, then we can try to do the inverse process and go from observed data and try to infer the causal structure underlying our variables. Just one slide with some formality. We use here graphical models to represent causal structures. Graphical models like this combine the rigor of the probabilistic approach. Why? Well, because we can read out the conditional probabilities that we expect with the intuitive representation of relationship given by graph. It's easy to see that here x1 has a causal effect on y and no y has a causal effect on x1. So last year, actually, we proposed a methodology to reconstruct networks integrating what we call multiple layers of omics information. So in this case, we propose to integrate genomic data, transcriptomic data, and phenotype data. But this methodology, it's easily extended if we have epigenomics data, if we have metabolite, or we have proteins. So the first step is basically to explore marginal association between the genotypes and the what we call endpoint phenotypes. That could be a phenotype. Then explore again marginal associations between genotypes and what we call now intermediate phenotypes. A classical intermediate phenotypes could be gene expression. And then we go to those genomic regions where multiple significant hits co-localize. I will show you in the next slide. And then we try to integrate that information and try to infer the network underlying this set of variables. Briefly, let me show you this example. We work with a unique pig population from Michigan State University where we have F2 pigs with phenotypic, genomic, and transcriptomic data. In fact, we have several phenotypes. We have genotypes. In this case, we use microsatellites, old genetic school, but easily you can work SNPs. And we use gene expression for almost 20,000 transcript measuring the loin muscle. In this case, we use microarray data. You can use count from RNA-seq, no issue. This is, I call, a unique population because we have almost 200 individuals, each individuals, several phenotypes, genotypes, and gene expression at the loin muscle. So as I mentioned before, the first step is to evaluate marginal association between genotype and phenotype. As you can see here, we found one genomic region in chromosome 6 that is significantly associated with 3 relevant phenotypes related with the muscle and carcass fat. And the second step was actually to perform another QTL mapping, but now using the expression as a response variable, this typically eQTL. And here I'm showing the result that we got in this region. And as you can see here, in this region in chromosome 6, we detect 7 significant eQTLs. And these 7 significant eQTLs are associated with the expression of 7 significant genes. So in the first 2 steps, we identify a genomic region in chromosome 6 significantly associated with 3 phenotypes, and at the same time with the expression of 7 genes. So the question is, can we infer actually the functional relationship between these 10 traits? We have 3 phenotypes and 7 expression traits. And the answer is, yes, we can. So here we have the graphical model that we got using, in red, the most significant marker in that region. We have 7 expression traits in yellow, and we have the 3 phenotype in blue. This is what we call a full direct cyclic graph because we could solve the direction of all the links. And if we see this graph, we can see that the genotype is marginally associated with all the expression traits. You can follow the arrows, and at the same time is marginally associated with the 3 phenotype traits. And these results are in agreement with what we'll discover before in the QTL analysis. So we were really glad with that. Just to mention that causal inference actually is like a 2-step approach. We first infer the structure, and after we know the structure, we can infer the causal parameters. So basically, the causal parameters are to estimate the causal effects. Some are positive, some are negative, but it's quite important. The third thing that we did is we tried to evaluate the stability of the network. So you present a result, and someone will come up and say, hey, so how precise are your results? How reliable is this network that you are trying to infer de novo? So we approach this method using resampling methods. In this case, we use jackknife resampling in order to evaluate the stability of the links between the variables, the stability between the link and the stability between the direction. As I mentioned at the beginning, if we know the causal relationship among the variables, we can use this information in order to predict the behavior of complex systems, something that is crucial for our workshop today. And this is really cool. So based on our network, we predict that the modulation of this gene, CNF24, should lead to changes to this gene, SSX2IP. CNF24 is actually a transcription factor. It's a thin finger transcription factor that plays crucial roles in cell differentiation and cell proliferation. A recent study actually wanted to investigate the target genes of this transcription factor. And this author, this smart author, what they did is they either overexpress this transcription factor or they silence this transcription factor and then they interrogate the transcriptomic in order to detect target genes. And look at the result that they got. Actually, when they overexpress this transcription factor, they found a decrease in the expression of this gene, as we predict in our network. And at the same time, when they actually silence this transcription factor, the silence of this transcription factor resulted in our expression of this gene, as we predict in our network. So this interventional study, and we call interventional study because actually when you work on this, you need to have a friend with a lab that can go and actually touch one variable in your system. These guys did this. They touched this gene. And the result of this study actually validate our network. But what is more important for today is we can use these networks that we infer from de novo and use that information in order to predict the behavior of these systems. So my last slide, I have some general conclusions here. We think that these gene set tools can greatly complement genome-wide association studies. Actually, they can enrich our interpretations. They can bring biology to a classical GWAS where we have some SNPs or even some candidate genes and try to put genes together in some gene sets. At the same time, we believe that we can use this gene set analysis beyond a post-GWAS, and we try to incorporate that information in prediction per se. In this context, we believe that the multiple kernel is a natural approach to incorporate biological information into prediction models. There are other ways to do it. We can use weighted G-matrix, but the kernel approach, using multiple kernel, it seems a really nice approach. Based on our studies, it seems that sometimes if we have a problem with the dimension of the data, really high dimensional data, pre-filtering the SNP using this simple concept of a marginal association between the SNP and the phenotype can reduce the dimensionality of the data, and we can improve the predictability of our models. And as you showed before, I hope I could convince you, this new approach of using graphical models and this coming multi-omics data try to integrate this, going beyond correlation and actually trying to infer the causal relationship between the variables. If we can do it, then we can predict really easily the behavior of these systems. With that, I want to acknowledge some guys. I want to acknowledge my group here in the University of Florida. I want to acknowledge Yi Han as a graduate student who has been leading the gene set bull fertility study. Rostan Abdullahi. Rostan is assistant professor in Iran, and he's a visiting scholar in my lab right now, and we are working together in this idea of multiple kernel whole genome prediction. Gota Morota in University of Nebraska, we have been working in gene set analysis, mainly mesh analysis in the last year, and the group in Michigan State University and in Wisconsin that we work together in this multi-omics causal inference project. With that, thanks for your attention. I'm open for questions.
Questions? Anyone?
Yes?
I don't think you said how the kernels were determined, which set belongs to which kernel.
Okay.
Can you repeat the question just so that...
So the question is in the multiple kernel. Good. In the multiple kernel, so we are fitting. So far in our models, we are fitting two kernels that are additive kernels based on the genomic relationship matrix. Now, the sum of the size of the two kernels, it's always the same. We use all the SNP that we have in the data. So here, for example, in the genic part, we use a kernel with the 26,000 SNPs that we call the genic SNPs, and then we use another kernel that we call the polygenic, that's the total minus the 26,000 SNP. Someone will argue and say, hey, Francisco, why don't you use one kernel using the 54,000 SNP, and then you count twice, and you incorporate another kernel? This is under discussion. We believe that this approach is better in order to separate and capture different contributions to the prediction of the model.
Thanks.
There's a follow-up question to that coming online. Did you try to then split the contribution of each kernel on the phenotype?
Yes.
Separated by...
Yes, we try. I don't have the result here. Here is obvious, for example, that this predictive ability in the multiple kernel is actually driven by the kernel that used just the significant SNPs. And in this case, we can see that in the multiple kernel, actually we improve the predictive ability of the model when we incorporate what we call the polygenic. That in this case, if we have 54,000 SNPs, and we're using only 1,000 SNP that we call Reaper SNPs, the other kernel has 53,000 SNPs. Yes, Jean-Luc.
A methodological question. On the top, with the
Yes.
13,000 SNPs, so these box plots are cross-validation box plots, right?
Yes. So this box plot, good point. This box plot, we have 5-fold cross-validation, and we replicate 5 times, so we have 25 points. This box plot are made with 25 points. Yes.
The 13,000 SNPs, do you ascertain them on the complete data set, or do you ascertain them on the trained folds?
No, that's a good question. In the whole data set. Good point. Only in the whole data set. So that's a good point. Probably we should do like this dirty GWAS in the training and then go to the testing. So Rostan is here, probably is taking notes, and we will start this afternoon. Yes, yes. All these ideas take time, eh? I mean, you need computational power and time to run. But yes, that's a good point.
So there's another online question as to whether you could or you try to assign functional categories to the SNPs rather than the genes. And I imagine you're referring to stop codon as well.
Yes. So far, yes. This is what I mentioned. So far, we work with genic, non-genic. What we are doing right now is actually split the genic within the gene or outside the gene, regulatory regions, and maybe we will explore exons, introns. Now, in this case, we don't have high SNP data. So we we are working here with 26,000 SNPs. So if we start to split the SNP in several categories, probably, I'm not sure how this will work. Probably that could be good if we have the 700,000 SNP in Catalan, we can work with that. Yeah. Yes?
Could you mix information, like including both information if it's genic or not genic, and information on the genome model?
That's wonderful, yes. This is the next step, is to go beyond these 2 kernels and have 3 or 4 kernels. And actually, you can think that you can have a kernel for geo-term reproduction, and then you can have another kernel for some mesh, and then you can incorporate in some way the marginal associations. Yes?
You can count SNPs twice, or you have to wait?
That's a good point. So far, we are not trying to count twice. This is why we have 54,000 SNPs, and we split. So if the SNP is here, it's not there. Now, when you start to making kernels with, let's say, 2 terms, one for reproduction, one for spermatogenesis, probably are genes that are in both, so you will be counting twice the gene. But you have to do that. So in that case, probably you will be counting a SNP twice. Yes, but so far, no. So far, it's black and white. Yes, Jean-Luc.
I mean, I appreciated the part of your talk in terms of inferring the causal network.
Yes, sir.
Have you tried to use that for prediction, or is that mostly for biological understanding?
So good point. So this one. So this data is, let's say, with microsatellites and microarrays, more like a toy example that we can do things that we propose. So this idea of this author that they predict the behavior of the gene is something that we predict in the network. Well, we'll say it's one gene, what is going on? Okay, now, but there are some people that is doing this at the level of the SNPs. So they create graphical model at the level of the SNPs, and then they use that information in prediction. Why does, if you want to predict this, for example, you don't need to incorporate this information, because given this is irrelevant. So using this is really nice to actually reduce the dimensionality of the data, and then you use less number of predictors in your model. The concept is quite easy. The thing is how you can go from observational data to construct these networks and then see the prediction. Yes?
So regarding these graphs, right? So we use some penalized likelihood to pick the best graph?
Say again?
Some penalized likelihood method?
No.
What do you use?
So basically we use conditional independencies, and the idea here is quite simple, is the conditional independencies that you observe in your data, in this case 11 variables, 3 phenotypes, 7 expression traits, and the genotype. The conditional independencies that you observe, those conditional independencies should be compatible with the model that you are constructing. And this uses an IC algorithm, typical from causal learning. Yes.
Does it have multiple possible outcomes?
That's a good point. That's a really good point. What he's asking is, what happens if you have one data that can generate conditional independencies, and that conditional independencies are compatible with multiple models? That's a good point. This is what we call statistically equivalent models, and this is something that with stat we cannot solve. So you go there and you cannot move. Fortunately, in this case, we don't have any statistically equivalent models. But this is a really good point. Sometimes you go and instead of have one causal structure, you have what we call a class of causal structure, that class actually produce the same set of conditional independencies. That means if you have the set of conditional independencies, actually you can go to the class, but you cannot distinguish between one or another one. Yeah. Good point.
Okay, thank you. Thank you, President.
Thanks.
I just want to make a quick announcement for the folks on the online streaming. If you have any questions, you can use the hashtag PPW16, so it's Phenotype Prediction Workshop 16. And time permitting, we're going to read your question here. Thank you.
So it's a great pleasure to introduce you to Jean-Luc Yannick. I think everybody that has worked in phenotype prediction recognizes him and his lab as probably one of the leading groups in addressing a lot of the questions that, especially in the early days of phenotype prediction model construction, many of us had. So it's really, he's been a pioneer in using this method, particularly in plant breeding. Jean-Luc, he got his PhD at the University of Minnesota. From there, that was in traditional breeding, and from there he went to Wageningen, where he got introduced, I guess, to molecular data. And then finally he took a faculty position at Iowa State, but he was in oat breeding, which I guess he tells me is not perhaps as...
It's not a terribly important crop in Iowa anymore. So.
Maybe he doesn't want to say. Anyway, he then decided to move, after he got tenure, he decided to move to Cornell University through a position that he had with the USDA as a research scientist. So thank you. We've tried to bring you here a couple of times ago, and so I'm glad that we managed to.
I'm very glad to be here. Thanks for persisting in inviting me. Thank you for the introduction. Also, before I launch into my talk, I want to thank Pancho because you'll see that there's some uncanny parallels between what he discussed and what I'm going to discuss. So he's done a great job of introducing my topic also. Let's see. So genomic prediction meets transcriptomics. In my case, I'm doing this SNP subset selection based on a separate transcriptomic experiment. And this is in cassava, so Manihot esculenta, manioc. I'll discuss it further. There are many authors. I'll give you a full rundown of contributions, but really I'm here to present mainly the work of Roberto Lozano and Dunia Pino del Carpio. It's a very big project, and I'll tell you all about our partners at the end. So cassava, it's a tropical crop. Both its roots and leaves are edible. So it's a very important staple in sub-Saharan Africa. Actually, hundreds of millions of people rely on it for subsistence. And half of it is fully produced by small farmers. So it's a very important food security crop. From a genetic perspective, it's outcrossing, so highly heterozygous, and it's clonally propagated. So you may have many generations of clonal propagation before you have sexual reproduction, and that means that it can have a very high mutation rate, so it's really highly heterozygous. It was actually domesticated in Brazil, in the Amazon basin, long ago, but it's been dispersed pan-globally in the tropics. Here, let me just show you as evidence. So this is the sort of importance of cassava on how many hectares of cassava are produced per kilometer squared. It's very important in sub-Saharan Africa, but also now increasingly in Southeast Asia. And so in part because of that, production in cassava is increasing dramatically recently, on par with maize, which is rather incredible. So cassava brown streak disease, it's caused by the cassava brown streak virus, and this is what I'm primarily going to be talking about today, is using transcriptomics to try and identify causal relationships among individuals. So just a little bit about that. It's not actually a terribly new disease. It's existed throughout the 20th century, but it was confined to the eastern coast of Africa, and it's only in the last 20 years or so that it has moved up in altitude and is now fairly prevalent in Uganda, where you can see cassava is a very important staple crop. And what it does is, this is healthy cassava. You have pure white starch. The root is really very high in starch content. The brown streak virus causes these discolorations, and it changes the whole, I mean, it's completely inedible. I mean, even goats won't eat this stuff. So the crop becomes unmarketable and is basically lost. It also has leaf symptoms. And of course, the scary part here is that while for now it's still confined to Eastern Africa, Nigeria, it doesn't exist in Nigeria, but Nigeria is very heavily dependent on cassava. And so we need to be very proactive in terms of trying to breed for increased cassava brown streak virus resistance in Western Africa, even though the disease is not there yet. It's transmitted by whiteflies, and so there is a clear path for it to get to Nigeria, and we believe that it's probably on the move. Okay, so we want to breed cassava, and we want to accelerate breeding. The main problem for cassava breeding is that it's clonally propagated. You create these cuttings from the stem, and one plant can produce, give or take, 10 of these cuttings. So the multiplication rate is slow, and I won't go through this whole set of trials that you would do to identify good clones, but it's basically only after 6 or 7 years that you're going to decide, okay, this clone is a really good one, and I'm going to use it as a parent in the next generation. We want to truncate that and be able to collect DNA on seedlings and then directly decide, okay, after 2 years possibly, move them on to back to a crossing block and greatly accelerate the breeding cycle. So just backing up relative to what Pancho presented, brief slide on genomic prediction. You have what's alternately called a trained population or a calibration population. It has genotypes and phenotypes. Then you come along with, there are many different names for these populations: validation, a test set, a selection candidates, or the selection target. These may only have genotypes, or possibly they have some low level of phenotype data, but not a full suite of phenotype data. And you're going to use the information that you have from your training population to calculate what we call genomic estimated breeding values, and you'll make selection decisions based on that. So you can do that as soon as you have DNA. And also now a brief introduction to GBLUP, which is the primary method that Pancho was talking about. How does it work? It's basically an information sharing method. Okay, so you have information on your training population. You don't have all that much information on your selection candidates. You're going to use this statistical technique to share information from one population to the next, or from one individual to the next. This works by means of a G matrix or a K matrix. In plants, the K has sort of become more popular as a designator of the relationship. The kinship matrix, that's where the K comes from. And so just to say what K is, it's actually a theoretical construct. It indicates how each individual should resemble the other individuals, okay? We don't know exactly how they're going to resemble each other, but this should comes from theory, and in particular from the idea of, from additive theory, from the notion that individuals that share single alleles, because parents transmit single alleles to their offspring, if they share alleles, they should phenotypically resemble each other. So again, information sharing. One individual informs the estimate of the other individual, and in particular, it's a form of constrained estimation. Okay, so the information that you have on one individual constrains the parameter estimates that you're going to get for the next individual. So just a brief statistical model here. So here are our phenotypes. We have some fixed effects, but really what matters here are these random effects, the genomic estimated breeding values, and they co-vary according to this relationship matrix, okay, this kernel. So what I've talked about here is a relationship matrix. Now we have SNPs, and we can estimate a relationship matrix, But really what we would want to know is the causal relationship matrix. And there's a difference between the two, right? So the causal relationship matrix is due to QTL alleles that cause the phenotype. The estimated relationship is generally a genome-wide sort of genome-wide relationship between individuals. But all of these SNPs, they're usually anonymous SNPs. We don't know if they have any effects themselves. So we're actually using this estimated relationship as a proxy for the causal relationship. Now, the difference between the estimated relationship and the causal relationship limits the accuracy that you can get out of these prediction models. And so some nice work by Gustavo de los Campos showed how that works. You can regress the estimated on the truth. If you knew the truth, this is sort of a conceptual idea. So you get a regression parameter, this b here. It's b goes between 1 and 0 and 1. And so the amount of variation that you could explain if you had the true causal relationship, you multiply that by this factor that gets smaller as b gets smaller, okay? And the point is that sometimes b can be quite small. So this was an example that he gave in that paper where, okay, so there are some individuals that in this data set that are strongly related to each other, but the vast majority of the individuals in the data set are very distantly related, and they cause this regression parameter to be quite low. I mean, you can see the one-to-one relationship would be something like this. So that would be b equals 1. Here it's equal to maybe 0.1. And by the time you square it, et cetera, the amount of variation that you can explain based on this estimated relationship is quite small. So what we want to do is, by this subset selection, I think this is actually what Pancho was talking about, is get at the genes that make up the causal relationship matrix. All right. So on the one hand, you want to identify the genes that make up the causal relationship matrix. On the other hand, to truly estimate that matrix, you would need to know how much variation each gene causes. And so I haven't talked about this perspective on it, but we're using SNPs as predictors. So each SNP has a regression effect. But because there are so many, we treat them as random effects. And so their effects are shrunken towards 0. Now, if you have a true causal gene, you don't necessarily want to shrink its effect towards 0. You want to give it as much leeway as you can. And I think this is exactly what Pancho is also getting at with his subsets. So you want to reduce the severity of the shrinkage by trying to create kernels, relationship matrices, that are a function of a smaller number of SNPs. The smaller number of SNPs allows each SNP to be less shrunken. And so this idea was put forward, among others, by Alk's Price Lab, Gusev et al. And just as a motivator, here's a graph from that paper. They did functional splitting among SNPs. So they have SNPs that are in coding regions, untranslated regions, promoters, DNA's hypersensitive regions of the genome, intronic inside genes, and then intergenic, so SNPs that are just far away from any gene in the genome. And the idea is that the rectangles that are in dotted line here represent how much variation you would expect these categories of SNP to explain if all of the SNPs explained equal amount of variance. And then the blue rectangles are how much variation they actually explain. And so you can see, just looking at the DNA's hypersensitive SNPs, that you would want to shrink the effects of these SNPs less than based on for example, for the intronic or the intergenic SNPs. How do you do that? Well, you use a multi-kernel model, right? So we even discussed. It looks like we even discussed the parameter values that we would use. But anyway, this is the same model as I showed earlier, except that now instead of having a single random effect for the overall value, I have in this example 3, and they each have their own separate relationship matrix. All right. So a nice thing about that paper, they also showed the need for sequence level imputation. So they did some simulations where they simulated effects for all of these SNPs, but then they tried to parameterize them only with, they had about a million genotype SNPs. So you know, I mean, this is humans, right? They have lots of genetic resources, but so a million genotype SNPs. But even then, with the level of LD decay, some of these SNPs, they're not actually getting at the causal SNP. And so the causal effect is in LD with some intronic SNP, and therefore it's being captured. Actually, the reason why this doesn't work so well is for the same kind of conditional independence reasons that Pancho was presenting. If you actually have the causal SNP, then conditional on fitting that causal SNP in the model, all of the other SNPs are completely independent of the model. And so all of the other SNPs then don't explain variation that is actually due to the causal SNP. So ideally, you want to have the causal SNP in the model. And to get the causal SNP in the model, you have to do, you know, you have to get all of the variants in the model that you can. So this is the overall framework that we're going to be working from, is multi-kernel models, trying to use genome-wide imputed SNPs. So just a little bit about all of the data sets. The field data comes from Uganda. We have about 1,000 clones. It's a little bit more complicated than I'm letting on here. These 1,000 clones are split into 2 panels. They were all evaluated in Namulonge, but about half of them were evaluated in Kamuli and Serere, the other half in Ngeta and Kasese. But so give or take. And then we're looking at leaf symptoms 6 months after planting for this cassava brown streak disease. So we don't have the money to whole genome sequence all 1,000, so we've been using this genotyping by sequencing, about $25 a pop. It's a fairly standard approach. I won't go through all the details here, but it leaves us with, before a whole lot of quality control filtering, about 155,000 loci. Whole genome sequencing, I'm not going to present a whole lot about it, but it really comes from Ed Buckler's lab. And so they sequenced, I think at about, 30x a number of cassava clones. 203 of them are from elite breeding, so that's the class of clones that are in our data. Again, fairly standard bioinformatic approaches. In the elites that matter to us, there's about 12 million variants that are identified. Okay, so I'm not sure how well you can see this. We just took the picture from the board, but we basically did this whole genome imputation in 2 stages. I'm not sure if that was the right approach. We're actually going to try a couple different approaches to see what works best. But we start with our genotyping by sequencing data, and we filter. So actually from the 155, we end up with 42,000 based on the predicted quality of the SNP and the minor allele frequency. Then we imputed first, where we used windows to avoid trying to impute too many SNPs in the same window. And that gets us up to about 700,000 SNPs. And then a second full imputation that gets us up to 2 million SNPs. The full 12 million that I showed, basically a lot of them get lost from quality filtering. And after the International Conference on Quantitative Genetics, at least in my lab, this whole WOMBAT issue has become quite popular. So WOMBAT stands for waste of money, brains, and time. And we weren't sure whether this would be one big wombat. But so that's what an artist in my lab, I guess, wrote up. Okay, so an important question here is, again, we're trying to tag these causal loci. And yes, the whole genome imputation does improve our ability to tag genes, at least. So from genome sequence, we know that cassava has, give or take, 33,000 genes. We're able to tag less than a third of them with a GBS marker set. But by the time we've imputed up to 2 million markers, we can tag a little bit over 2/3 of them. So there's still a fair amount missing. But we're doing much better. And also, we just have a lot more genes within a lot more SNPs within those genes. Pancho showed a number of graphs that are exactly like this one. This is sort of setting the paradigm for my talk on how I'm going to present results. So we have this 6 months after planting leaf symptoms of cassava brown streak disease, and we do 5-fold cross-validation. So each box plot is representing actually 25 points. And this is the accuracy that we get from genotyping with the genotyping by sequencing data, with the first stage of whole genome imputation and the second stage. Now, okay, so the jump isn't huge, like 3 percentage points, but it is significant. And one thing that we do know is that this isn't caused by increasing the number of SNPs. We know that with 61,000 SNPs, we have plenty of SNPs to get good accuracies. So actually what we think is happening is that this whole genome imputation is actually working as a form of quality control. And this is a GWAS plot with whole genome imputed SNPs. This is just on marker 4. And I'll get back to the fact that this is a big QTL. It's smeared across almost the whole chromosome. But another thing that you notice here is that we have these sort of pillars with kind of gaps in between where there aren't any SNPs. And so this is sort of, this is what we think is going on in the filtering process. Some of these regions just don't have high quality GBS SNPs that allow us to impute. And the lack of high quality GBS SNPs is, well, the removal of some low quality GBS SNPs is probably what is leading to this boost. So first we do a GWAS, and basically there are two big QTL, one on chromosome 4, one on chromosome 11. Cassava has 18 chromosomes. Yep, so there are two QTL. And we do have biological hypotheses for this. In particular, this chromosome 4 QTL, when both cassava mosaic disease, which is another viral disease in cassava, and cassava brown streak, initially they tried to bring in some resistance to these diseases by crossing with congenerics, so related species of cassava. And so this is a visual graphical genotypes. Each row represents a clone, and each column is a SNP. Again, chromosomes going from 1 to 18. But you can see in chromosome 4, there's this big segment that comes from cassava glaziovia, manihot glaziovii. And so that's the dark green segments are pieces of an alternate species. And it's segregating in the population, and we think that that's causing this big QTL there. On chromosome 11, the QTL there, these are the GWAS hits. There's some differential expression that we show here, but in particular here there are a number of NBS-LRR genes. These are resistance gene analogs, and that coincide with the peak. So we think maybe that's why this is happening. So again, very similar to what Pancho showed, we can create a multiple kernel model. Here we have a kernel that's due to chromosome 11, a kernel that's due to chromosome 4. Then we pick 2 chromosomes at random, not 11 or 4, and we make kernels out of them. And lo and behold, they explain less variation, or they generate less accuracy than the kernels from chromosome 4 and 11. And we can sum up the effect of 11 and 4 and get something that's almost as good as the whole genome. Whereas if you sum up the predictions from the 2 random chromosomes, you get much less. In fact, this was so good that we wondered, okay, is 11 and 4, is that the whole thing, right? Is there anything left in the rest of the genome? Do we need to worry about the rest of the genome? So we did a 3-kernel model. So EE is everything else. So in red here is now a 2-kernel model with just 11 and 4 and their sum. And in green, you have 11 in the presence of a kernel that incorporates the other 16 chromosomes. And again, 4 is the chromosome 4 kernel in the presence of a 3rd kernel that has the other 16 chromosomes. And, well, this kernel is highly significant. There is significant other variation in the rest of the genome. And in fact, what we think is happening is actually chromosome 11, it has, I mean, any chromosome is part of the same organism. So there is some similarity in the relationship between chromosomes 11 in all of the individuals and the whole genome in all of the individuals. Okay, so when you fit this kernel, it removes all of the similarity that chromosome 11 was accounting for, that was just based on sort of its similarity by descent to what the rest of the genome was doing. Chromosome 4, so in other words, there's a big drop in accuracy for chromosome 11 when you go from a kernel without the everything else to the kernel in the presence of everything else. Chromosome 4, not so much, and chromosome 4 actually has a kind of relationship matrix that's all its own because of this funny big segment from another species. Okay, so that's what we can do based on GWAS. Now we're going to try and pick subsets of SNPs based on transcriptomics. So this is the transcriptomic experiment. It has these aren't individuals that were measured in the field, so this is a completely separate data set from what I've shown you so far. There is a susceptible clone, Albert, and a resistant clone, Namikonga. They were either, so you infect them by grafting them onto an infected rootstock. And so you can either graft them onto an infected root, or you can graft them onto a root that's not infected, so a mock inoculation, right? And then you pick RNA, 6 hours, 24 hours, 2 days, 5 days, 8 days, 45 days, 54 days, and you do RNA-seq. So it's a pretty standard RNA-seq pipeline. I guess I won't go into it, but basically, sequencing, quality control, aligning with the genome, and then quantification. If you want to talk to me, actually, if you really want to know about it, you're going to have to talk to Roberto Lozano about it. So one of the things that's interesting is Cassava-Brunswick disease is actually an RNA virus, and it does have a poly-A tail. So when you do RNA-seq, you also capture viral particles. So here, the top row of graphs is the susceptible clone. The bottom row of graphs is the more resistant clone. This first column is the one that was mock inoculated, and the second column is the one that was truly inoculated with the virus. And on the x-axis is just a quarter of a day on up to 54 days. And so the thing that we were a little nervous about, but hopefully it's okay, but I guess maybe we should talk to more plant pathologists about this, but there is some virus in the mock inoculated clones. It's about 1,000-fold less than in the inoculated clones, so hopefully it's not having a big impact on on the experiment. But anyway, it's a good way to verify that. Also, you can see that the resistant clone has about 200-fold less virus titer than the susceptible clone. All right, so then we look at differentially expressed genes. So here, these are just like the result of t-tests between pairs of samplings, either in the susceptible or in the resistant. And you can see the numbers of differentially expressed genes in red as you go along. We were actually kind of nervous about this. No differentially expressed genes. I mean, you'd expect, at least from noise or something, that you'd find some differentially expressed genes. We would expect, we hope that there's a sort of reason for this, as opposed to, our own empirical failures, that the susceptible clone is basically unaware that it's being attacked by the virus, right? And so it's just continuing on with its usual program of gene expression, and there's not much difference then between inoculated or not inoculated clones. And then at a certain point, it starts to get really sick. And when it starts getting sick, then there's differential gene expression based on it being sick. On the other hand, the resistant clone is, sort of figures out to anthropomorphize that it's being attacked by a virus and sets in motion a whole resistance pathway. So so then the question is, okay, so how are we going to use this gene expression data to select a subset of SNPs? And we decided to go with genes, that that show a genotype by inoculation interaction. Okay, so there's no obviously there's genes that are differentially expressed between the two clones, but there are many genes that have nothing to do with disease resistance. There are also genes that are going to differ in their expression based on inoculated versus mock inoculated. Those, in principle, don't differentiate the resistant from the susceptible clone, at least not as much. So anyway, the pattern of differential expression that we decided to look at was this. And so you can make a sort of differential expression Manhattan plot. Now there are 33,000, or actually about 10% of the genes weren't captured by RNA-seq. So 10% of the annotated genes. So it's not quite 33,000 dots, but anyway, each dot is a gene, and the negative log 10 p-value is the p-value for this genotype by inoculation interaction. And I guess I didn't know what to expect. This sort of surprised me. It seems fairly uniformly distributed across the genome, right? I was sort of hoping that maybe we'd have a big concentration on chromosome 4 and on chromosome 11 or something, but okay, this is what we get. Happily, and so now we're doing a very similar thing, but instead of doing it chromosome by chromosome, well, there's a chromosome component to this, but we're going to make kernels with differentially expressed genes versus kernels that have the same number of genes, but these genes are picked at random as opposed to being differentially expressed genes. So we identify the genes, we pick all of the SNPs that are actually inside the gene, and those are the SNPs that we make our relationship matrix out of. And again, we do five-fold cross-validation here. So this is chromosome 11, all differentially expressed genes on chromosome 11. You make a relationship matrix out of that. This is what you get. This is what you get if you pick random genes. Same thing for chromosome 4. And this is the differentially expressed genes everywhere else on the genome. And basically, in each case, this green box plot is a little bit higher up than the blue box plot. The blue box plot, by the way, has larger variance because each time we do a cross-validation, we pick a new random subset of genes. So there's some variation that's due to picking random subsets of genes. And also, if you sum up the effects of each of these three kernels, you get higher accuracy. I mean, it's not dramatically higher. I'm not saying that we're going to revolutionize plant breeding with this. But anyway, you do a better job with the differentially expressed genes than with the random genes. So we were happy about that. Right, and similar to something Pancho showed, here we're working with just like about 3% of the SNPs as opposed to all of the SNPs, and we're getting a similar accuracy. So then what I showed you before here, these are kernels that are built only with the differential expression. What about the rest of the polygenic component? So what's new here is now I'm comparing a 3-kernel model to a 4-kernel model. These are the 3 kernels are all constructed with differentially expressed genes, but we've added for the 4-kernel model a 4th kernel that has genes from all of the other SNPs. That they have to be at least 5 kb away from a gene that is differentially expressed. Okay, okay. So what you find is that I'm not sure if I'm overinterpreting, but there's a pretty strong decline in the accuracy of the kernel. The accuracy that's generated by this kernel that has differentially expressed genes on all the other chromosomes. Okay, so just to make sure you understand the nomenclature, the blue is the 4-kernel model that has this kernel that's the not differentially expressed kernel, and so when there's a plus, that just means it's the 4-kernel model. Whereas for chromosomes 11 and 4, adding, and here we're adding 1 1.9 million SNPs. I mean, there's a kernel that really has lots of information about the genome in it, but there's not a big decrease in the accuracy that's generated by these kernels on chromosome 11 and 4. So I don't know if I'm overinterpreting, but the interpretation that comes to mind is that these large effect QTL, they are caused by differentially expressed genes, and so their effect is captured by differentially expressed genes. On the other hand, possibly a good fraction of the polygenic component is not due to differentially expressed genes, but due to other functional reasons. So that when you add this kernel, it's capturing the non-differentially expressed component and causing this drop in accuracy. All right, so that's my last data slide. Oh no, one more point, and this is a point of disappointment. Okay, so we've tried to be clever. We've tried to identify these subsets of genes, and we have kernels that are much more focused on genes that we think are relevant. So we're causing the SNP effects at these SNPs to be less shrunken. We were hoping that would actually lead to an increase in prediction accuracy. And I mean, if you're very biased in favor of all of these approaches, as I am, you can say, well, maybe this box plot is a little bit higher than this box plot. But basically, the ultimate goal that I was going for of increasing prediction accuracy hasn't come about, and I'm not entirely sure why that is. I mean, I agree with Pancho that multi-kernel model is a very natural way, intuitively, statistically, whatever, of incorporating biological information, but we've put a lot of effort into this, and meh. Okay, so here we go, some tentative conclusions. We think that really this whole genome sequencing and imputation is not a wombat. Like, it's valuable for us, and it is a significant investment of money, brains, and time. I'm not saying, but I think it's quite valuable. Pretty straightforward GWAS conclusions. We have some QTL on chromosomes 4 and 11, and they matter for prediction. Differentially expressed genes appear uniformly spread across the genome as opposed to focused in on these QTL. And they do explain more variation than just random genes. And then this is the tentative part of the conclusion. Maybe these variants in DE genes are really what are causing the major QTL more so than the smaller effect variants that are happening genome-wide where there's no major QTL. A couple of questions in case anybody in the audience wants to help me out in my research. I showed that we explored just one pattern of differential expression. Potentially there are many different patterns we could look at over time, over the different treatments that we have. Should we look at other patterns? This again goes back to this sort of note of disappointment is we think we're doing all the right things. Why isn't this increasing our prediction accuracy? And so then we're left with, okay, so in human genomics, of course, you can really get good, and I think in cattle genomics at this point, you can get really good imputation across the whole genome. Is that the level of genomic resources that we need to be able to nail down these kinds of phenomena? And with that, let me just move on to, so this is the talk, I don't think I've ever given a talk which sort of encompasses quite as much data as this talk before. As I mentioned, really, the two people who are most responsible for the direct analyses that I showed, all of these box plots, were Roberto and Dunya. But there's a lot of field work that goes into this. Genotyping by sequencing in itself is not trivial. Whole genome sequencing, several labs, Cornell, the Joint Genome Institute, Donald Danforth Center. And then there was a whole separate effort at doing RNA-seq. That was led by IATA, but with the help from Joint Genome, Dow AgroSciences, and Cornell, and me. I'm just, I'm encouraging everybody. I'm trying to help with interpretation, and I get to give the talk. And then, before you go away, let me thank all the other people, and particular, so this Cassava project is funded in part by UKAID, so it's the equivalent of USAID from Great Britain, and by the Bill and Melinda Gates Foundation. Cornell, I'm with USDA-ARS, the Boyce Thompson Institute, Joint Genome Institute, we're all partners. And then the really important people are in Africa, International Institute for Tropical Agriculture, the Nigerian National Program, which is the National Root Crops Research Institute, and in the national program in Uganda, the National Crop Resources Research Institute. Finally, if you were interested in this talk, know that we at Cornell have a quantitative genetics position that's open. It's a beautiful place to live, not quite as hot as down here. And if you like winter sports, it's awesome. And there's just fabulous colleagues. So with that, go ahead and ask questions, I guess. Am I on time? Do we have time for questions?
We have time for questions.
Okay.
Has the identification of these two chromosomes resulted in any breeding success?
Presumably, we haven't. I mean, this is really hot off the presses. I mean, we've known about there's other evidence for chromosome 4 and chromosome 11 from biparental mapping populations as opposed to GWAS. And so we're gearing up to do marker-assisted selection. So far in the efforts that we're doing in this Next-Gen Cassava project, they've focused entirely on genomic prediction as opposed to using a very much reduced subset of markers to quickly go after specific regions. But so no is the short answer, but hopefully within a couple of years, yes.
The history of this population is probably quite unique, right? You probably have severe bottlenecks. You have probably some level of maybe some inbreeding or maybe not. Do we know anything about LD? What it looks like?
Yeah, we do know about LD. It decays fairly rapidly in cassava. Cassava is quite diverse. I mean, much more diverse than other species I work on, like wheat and barley and oats, small grains in general. So to quantify that, I mean, LD decays to background levels in the broader African cassava population already within 20 kb or so. So anyway, it decays quite rapidly. Yeah.
The other thing related to the research in general is makes the general assumption that gene expression is a major driver of the phenotype. Right. How do you view the balance between these two things? First, genetic variants that impact the structure of a protein that impact the phenotype versus genetic variation that impacts expression of a gene and then impacts the phenotype.
Yeah. Well, I mean, I do think that the gene expression. So I think there's a lot of evidence now. I mean, that gene. Can I go backwards at this point? Yeah. I mean, well, so I don't know if I'll go all the way back in my talk, but that slide from the Elks Price group with the DNA's hypersensitive sites and how much variation they can explain. I mean, those are all promoter enhancer type locations as opposed to structural effects in the genes. So I mean, I think that also points to the expression being a major driver of the phenotype. Yeah.
The kernels that you built for the differential expression, were they using the differential expression measures, or were they SNPs within genes that were differentially expressed?
The SNPs within genes that were differentially expressed. Yeah.
Do you also have a measure of like amount of variability that just the absolute expression is explaining?
So, I mean, so the differential expression experiment was just between two, I mean, it doesn't, I mean, it would be a dream world if we could collect differential expression on the full set of 1,000 clones, but we don't have that. Now we could try to differentially weight the SNPs in the kernel based on the p-value, I suppose, or something like that of this interaction. We haven't tried that. The problem with this kind of research is that there's at least 100, 1,000 different ways that you can construct these kernels, and you're tempted to try them all. And one of the reasons why we limited ourselves to just this one pattern of differential gene expression was, you drive yourself crazy if you try too many different things. They're not orthogonal to each other. And so I'm not quite sure how this works out, but okay, so let me see. This does go back to the Alex Price research, where he showed that if this is it. If you actually have the causal gene or the causal SNP in the kernel, and in principle, conditional on fitting that causal SNP, the other SNPs are then independent of the phenotype. So whether the kernels, I mean, this is getting a little bit beyond, my understanding of how the statistics work, but whether or not the kernels are completely orthogonal, again, if you fit the causal gene in one kernel, then its effect should not be picked up by the other kernels. Do you have a comment on that, Pancho? I don't know. Yeah.
It's fine. I think when you see you have a kernel in chromosome 4 and a kernel in chromosome 11. I think when you sum up, you get, what is the variability that you get?
Well, okay, sorry, I'm roaming back and forth. I mean, it's less than the, well, okay, so this is just the two kernel model with 11 and 4. Obviously, the sum of the effects coming from here doesn't generate an accuracy that's equal to the sum of the accuracies. I mean, yeah. And actually, I mean, I've wondered. So we're showing the exact same kind of graph that you showed. Maybe we should just show the variance components that are estimated from these kernels. That might be a clearer.
Well, this is something that we have estimated with Rostam.
The variance components.
Yeah.
Yeah. I mean, we have the variance components too. It's just I don't know. I don't know why I decided.
So you don't expect that error tolerance, eh?
No.
Because you have a population structure behind that, even if you
Right. Exactly. Right. Paul.
Yeah, for the imputation to work well, you would hope to have a very good quality reference map. I'm curious on a crop like that, the quality, but also specifically on the chromosome 4 and 11. If you have foreign or from a
No, that's a very good
species
point. Yep.
DNA, then when you're trying to just do the alignment,
Yep.
it seems like you might only have the cassava DNA and not the foreign one.
Yeah.
Whether even the alignment is going to work properly.
That's a good question. So there is a decent reference sequence. It's not. So it's constructed both by traditional reference sequencing where you have contigs and scaffolds and so forth, and then there is a consensus genetic recombination map that was developed out of 10 biparental populations. And so the positioning of different scaffolds relative to each other is depending on this genetic map, so recombination between the scaffolds. But obviously or I don't know obviously, but the reference sequence doesn't have this segment of chromosome from Manihot-Glazieovii. So it's true that imputation there could well be a problem, and we should look at it. In general, I don't know exactly why, I mean, this is something we need to look in further, why there are these sort of gaps in the imputed genome.
But the inserted DNA at least has the same genes from a similar species?
Yes. I mean, I think it's not that different. I mean, they're sexually compatible, so yeah. I mean, I don't know exactly. Patricia?
When we reduce the number of SNPs, it's been shown that it can impact the long-term,
Right.
models.
Right.
In this case, you are using, the expression genes and they have a similar accuracy that the whole genome prediction.
Right.
It's better now, but what do you think is going to happen? I mean, these are not the causal variants, basically. 2LD, basically, in the model prediction in the future, you're going to have issues with it.
Okay, what Patricia was pointing out is that if you knew the causal genes and then the variants that they produce, then you can get a correct causal relationship between two individuals, no matter how distantly related they are. LD decays, et cetera, genomic phase or LD phase changes as relationships get further apart. So if you don't have the causal relationship matrix, the estimated relationship gets worse and worse as individuals get more distantly related. So I guess, one hope is that, another reason why we're chasing after this is that we do really want to, sorry, forget how many slides there are in this thing. We do really want to start making predictions for West African cassava, right, based on training populations in East Africa. It's very difficult to move cassava germplasm from one country to the next because people are scared silly about moving viruses around, right? We are usually botanical seeds, so the seeds of the cassava are virus-free. So what we are going to start to do, of course, seeds of an individual are not the same thing as the clone, the individual itself, but we're going to start sending seeds from clones in Nigeria to Uganda to get assessments on, so basically progeny tests, Nigerian clones for their resistance in Uganda. But we hope that getting closer to the causal relationship matrix will improve prediction accuracies for West Africa, so more distantly related material. I'm not sure if that answers your question, but...
Yes, but in the long term, the model is supposed to be used for several generations.
Right,
hopefully.
right.
So in the long term, if actually it's not the causal variant, basically due to the LD and the recombination, you're going to start breaking down the prediction ability of those markers, and then your model is going to suffer instead of using whole genome.
Well, the whole...
all the markers that you can.
So we have three situations here. We'd have a reduced number of SNPs, but they're causal. You have a reduced number of SNPs, and they're not causal. They're just in LD. Or you have the whole genome. So thinking about those three scenarios, I'd rather have the whole genome than the reduced number of SNPs non-causal. Now, I guess there's a little bit of an intermediate, which is that you have a reduced number of SNPs. They're not causal, but they're in tighter LD, or they're more closely linked to the causal SNPs. Yeah, I'm not quite sure how that'll all shake out. I guess we'll empirically try to figure it out.
Just regarding the gene expression study still, you have a single genotype that's resistant and a single one that is
Susceptible.
right? Assuming that there's a common effect that's shared among genotypes that are resistant or susceptible, well, particularly the resistant ones, would it be interesting perhaps to look at 3 or 4 different genotypes that are resistant and look at commonalities in the gene expression pattern relative to the susceptible ones?
Yeah, A, that would increase our statistical power for identifying differential expression. B, that would...
remove a lot of the noise that you have.
Yeah, that's true. Right, we are definitely going to be pursuing RNA-seq further, yeah.
What's the question all the way?
How do you know they were not contaminated with other viruses? They were tested negative?
Well, okay, so that was, we actually do know that they were slightly, the mock inoculated ones were slightly contaminated. Whether that came from white flies that were flitting around the greenhouse or, some other, some other kind of contamination. Here we go. So here, this left-hand column of plots, these are the mock inoculated individuals. And you can get from RNA-seq, you can get a virus load of, so on the y-axis, I should have said, is the log of the number of reads that come from the cassava brown streak virus, okay? So after 45 days, 54 days, you have some load of virus. There are different forms of contamination that are possible. So, okay, so this is the data that we have. It's good that we can verify what went on by using the same RNA-seq data, but it does mean that our data is not perfect. Yeah.
Okay. Well, thank you, Jean-Luc.
Thank you.
We're about to start. The next speaker is Dr. Arnie Wilkinson, something
Good.
like that. That's good. So he's a postdoctoral fellow in Aarhus University. Right now he's working in statistical and quantitative genetics models. He did his PhD in 2011 in computation biology in the University of Southern California, working with Dr. Magnus Norborg. After that, he moved to the Harvard University, where he worked with Alkes Price, working in similar projects. And since 2004 now, he's working as a postdoctoral fellow and focusing on statistical methods for prediction using for polygenic traits and also for disease risk assessment. So it's a pleasure to have you here.
Thank you very much for that very nice introduction, and thank you for inviting me. So I'm interested in polygenic prediction of human traits and disease risk, and sort of my background, as you mentioned a little bit, when I was the PhD student, I was actually working on plants, and I was doing plant genetics, and I read some of this breeding literature, and sort of realized that these are really experts in predicting things, right? They've been predicting traits since the 50s, right? The mixed model and these things that have now become omnipresent in human genetics, it sort of originates from animal breeding and plant breeding and so forth. So yeah, I wanted to sort of point that out. Now, in human genetics, prediction is no longer science fiction, right? I don't know, does this work? Yes. So in human genetics, they're sort of mostly focusing on predicting disease risk. There's also sort of other types of prediction, which I'll also talk about, but sort of it seems like they are mostly focusing on predicting disease risk, and genetic testing, is already sort of routinely used in clinical settings, for example, breast cancer, and this is sort of the Angelina Jolie effect, right? This is a stock of the company when this news broke, had the patent on the breast BRCA gene on the BRCA gene actually, I think, at the time, if that was in 2013. Right, anyways, in human genetics, genetic testing is really quite common nowadays in clinical settings. And I was thinking about this yesterday, so I think you can sort of split these, the utilities of these genetic testing into sort of three categories, right? The purposes, right? You can do them for preventive screening. Say, for example, you have a patient or an individual that has some family relatives that have breast cancer, and then you go and talk to your doctor, and well, there's some reason to believe that you might perhaps have some mutations that would increase the risk of you getting cancer, so you do a BRCA gene test, for example. Another reason for doing genetic testing is perhaps, for diagnostic purposes, right? If you have a patient, you don't really know what's going on, or perhaps you have a hypothesis that it is this particular condition that is governed by a mutation in a certain gene, for which there's a gene test. You can sort of either try to sequence patients if you have no clue what's going on. That happens actually quite often nowadays in sort of pediatric kids that are born with severe issues. Then you sort of try to figure out what's going on, and one way of doing that is sequencing them and just looking at sort of plausible causal genetic variants. You can also sort of try to verify your diagnosis for other things. And so a third case where genetic testing is being used in clinical settings is for drug efficacy prediction. For example, in cancer genetics, I think this is quite common there. You sort of sequence the cancer, and you try to figure out which drugs you can use to try to kill it, right? And those are sort of the three utilizations of genetic testing that I can think about in the clinic. I don't know if you have any more, but those are the three. So in recent years, right, genetic testing has become really common. And the reason for that is sort of twofold, at least. One is that sequencing and genotyping costs have just dropped dramatically, right? So now you can sequence an individual genome of an individual for $1,000 or so. And on top of that, if you don't need the entire genome and are sort of okay with getting more or less sort of 99% of the variation, you can probably make do with a chip, right? You know, a dense genotyping chip, which is even cheaper. You can get that for $100 or something, if you don't care about sort of the rare or new mutations, right? And you know, and you can sort of even, you know, if you want to get your genotype yourself, you can genotype yourself through many companies now, and look at that. And some companies even will pay you for your genotype. So in Iceland, I'm originally from Iceland, right? In Iceland, Decode Genetics actually went out and knocked on people's doors and sort of suggested that, you know, asked people to donate their DNA, and they would, you know, give $20 to a good cause in return in your name, right? So I think 50,000 people or so gave the DNA in that thing. So people are even, you know, companies are even going after your genetic data and want to use it in the research, right? Another thing that has sort of changed the sort of gene testing landscape is the myriad genetics decision, right? So the Supreme Court of the US had an important decision in February 2013 against myriad genetics that had the patent on the BRCA gene, right? That basically meant that you can't patent genes anymore. And that means, of course, now we're finally allowed to look at our own genomes, which is nice. But it also means cheaper genetic testing because of competition. Now, basically every sort of, if you're a major genetic testing company, first thing you do is implement the BRCA gene test, because that's one of the sort of most common tests that people do. Right, and these things together, right, the decision of the Supreme Court and cheap sequencing means that you can also come up with these sort of tests that try to do everything at once, right? So Illumina now, if you go to the web page, I think you pay $12,000 or $11,000, and they will sequence you, test 1,700 genes for risk variants for, you know, 1,200 conditions, and give you a detailed report, right? I think here this is sort of aimed at you know, patients for which you don't really know what's going on, and you sort of want to try to figure out what is actually causing, you know, the condition, right? And for example, Nextcode just went to the webpage yesterday. They offer the same pediatric rare disease diagnostic, which is sort of Illumina is aiming for with this, I believe. But they also have some preconception carrier screening and whole genome consumer wellness scan. And, you know, these are presumably, you know, serious companies, right? Be careful, this is being taped. Right. Good. Anyways, genetic testing is currently a hot topic, right? And you can see that in sort of statistics of the NIH Genetic Test Registry, which in the last 4 years has grown from being just, I think, 1,000 tests or something like that, to having 35,000 tests, right? And these are tests that I think, you know, test for more than 5,000 conditions, where they test, you know, 3,000 genes, and if you split them up, you see probably 10% of them were basically BRCA gene tests. And then you have, you know, various other single gene tests, like a quarter or something, some whole sequencing tests, exome test, genome test, mitochondrial. There are all kinds of tests there, right? From lots of different companies. So this is really popular. It's you know, people are willing to invest this money in this. So that sort of suggests that people actually believe that this is the future. a lot of people at least. Then there's also this other side of genetic testing, which is not really happening in the clinics, and that's sort of the direct-to-consumer genetic testing, right? You probably heard of this company, 23andMe. You might even have your genomes there, or Ancestry.com. And these companies sort of offer direct-to-consumer genetic testing, where basically they will genotype you and give you some information on various health things and health-related phenotypes, and also tell you sort of fun stuff like whether you like cilantro or not, or in case you didn't know. Right. And Ancestry Health, I haven't really looked at that very carefully, but they also do some, or plan to do some give you some health predictions basically. There were some legal issues. It seems like a lot of that has been sort of resolved basically by 23andMe now being more transparent in what they're actually doing, and also sort of toning down their claims, right? Which is sort of important, I guess. And it's not legal in all countries. In some countries it's legal. It's a bit of a mess, like whether this is legal or not, right? Recently they also proposed this direct-to-consumer genetic testing for researchers. Here the idea, I think this is actually an interesting idea. Here the idea is that researchers can sort of recruit patients or people, I guess, not necessarily patients, people to participate in studies through 23andMe, right? And that sort of in return gives researchers access to the genetic data and phenotypes, but in return it also gives individuals access to their own 23andMe data, and it also gives the researchers access to existing 23andMe data, so they can also use some other genotypes perhaps, and that basically allow you to sort of subsidize the genotyping, right? And which is then an incentive for you to actually participate in the study, right? And there are some potential other non-profit competitors in thinking about this, right? There's DNA Land, which is this thing that was set up in the New York Genome Center, and then there's this website that I've been working on for a while, and it's not very fancy, but we're still ongoing research, which I'll talk about later. Right. I think it's also incredibly important to realize that genetic testing has limitations, right? And sort of talk about them a little bit, right? First of all, right, they're expensive. A lot of these genetic tests are quite expensive. And a lot of these, genetic tests are perhaps single gene tests where you test for specific variants in a single gene or sequence a single gene. And even though you're just testing for a few variants, it costs like a few $100s. I don't know why, but apparently they do. Some of these tests, like the Illumina sequencing test, right, that cost approximately $10,000. There you also get a full sequencing of your genome, but you also get sort of a report, because for sort of the layman to understand a genome is pretty difficult, right? So the report is presumably probably a lot of the price. But it seems also there's a bit of a lack of standards for some of these types of tests, at least, and the regulation framework is sort of a little bit unclear. And in fact, I think I noticed that the FDA is now asking for comments on a draft to regulate sort of whole sequencing in clinical settings, and nobody has submitted any comments, and it's due October 6th. So I'm thinking, like, people here, you might want to sort of participate in actually suggesting, how they should do things. I'm actually considering I just don't have time like anybody else. Right? Anyways, these genetic tests may also be biased for certain groups, individuals of certain genetic ancestry, and I'll talk about that a little bit more. And it's also important to remember that sort of wrong diagnosis can have drastic consequences, right? So, we're talking about people. We're not talking about cattle where you can sort of cull animals, right? We're talking about people, and that's really important. There was an Atlantic Journal article December last year that pointed this out. Actually, this point that I talked about here, that they sort of can be biased to certain groups of perhaps individuals of genetic ancestry. What happened here? This is a story about a family. What happened here is that there was something that they observed in the fetus or something like that, and they thought it was a Noonan syndrome, a Noonan disease. I don't really know what that is, but they tested for that, and they found a genetic variant that was had been suggested to be causal for that syndrome, and they actually terminated the fetus based on that evidence. And later they found out that this genetic variant is quite common in Ashkenazi Jews and seems to be benign, and these were actually Ashkenazi Jews people, right? So that's quite sad, potentially, if that was a benign variant, right? And so this is a paper that just came out like last week that sort of highlights this, potential for health disparities like in genetic testing, and I think this is sort of important, right? That basically they're pointing out that some of these tests for hypertrophic cardiomyopathy, which I don't really know anything about, again, can be biased if you're African American, basically, and sort of talking about the potential prospect of that. So I'm just going to talk about an actual individual here, Jonas. I've never talked about this before, but I'll at least not here, but given I talked about this before, but I'll also talk about it. So Jonas was found to have a nodule thickness. Like if you ever had a kid, you know that they test the thickness of your neck at around 12 or 13 weeks, right? And if that can be an indicator of having Down syndrome, right? So Jonas was given a 50% chance of having Downs based on his thickness. So then I went on and did a genetic testing of placenta, which is actually consists of the same genetic material as the fetus, right? And they ruled down Down syndrome, right? But they found a large inversion on chromosome 2. So then I went and tested the parents and found that the inversion was actually segregating in the mother and therefore benign. Okay, so then I waited a little bit. 18 weeks, another potential reason for having nodule thickness is that you have some cardiological problems, right? So they looked at the, when it was tested, looked at it again and found some valve problems with the heart, right? Heart problems. The cardiologist, again, gave him about 50% chance of surviving the pregnancy. A week later, and this is true, the cardiologists were stunned to find that this Jonas's heart seemed now okay. So, but still, they were surprised by this, so they wanted to study him further. So there were some more ultrasounds. Then there was something wrong with his kidneys, perhaps. They were perhaps too big, and his legs too short, and so forth. And later, at week 28, the parents basically decided, they couldn't take these ultrasounds anymore. And if you didn't figure it out, this is actually my son I'm talking about. So it was a bit difficult. But he was born in Boston, and an interesting date, a lot of 13s. But he's fine. He's now running and talking and enjoying life as any normal 3-year-old. And follow-up screening found that there was nothing wrong with his heart. But I think this is sort of a nice example of, pointing out that risk prediction is difficult because it affects people, right? It sort of affects real people. So I think it's important to try to improve genetic testing, right? And I'm very motivated to do that. One of the things that you can do as well, get better training data. I think using more data, polygenic predictors that aren't just testing for a single gene, but testing for, looking at more variants in the genome is very important. And, breeders know that, right? That's actually what they've been doing for, decades, right? And these talks that we saw earlier, they were all sort of polygenic in some sense, right? I think also additional information such as environment, family is also important. And that's sort of, a lot of these things are already being used, right? If you go to a medical doctor, they ask you about your family, right? They're sort of implicitly, in using these things to try to get an idea of what your risk is for getting a certain disease. And the second thing that sort of I think is important in order to improve genetic testing is independent and thorough validation. I'll get back to that again. So it's important to use representative validation samples. So if you think a certain genetic variant is indicative of a disease, and you find that based on looking at Europeans, right? Then taking that into African Americans or people of different genetic ancestry may not always be the right thing to do. You should probably test it in population with similar genetic ancestry before you sort of use that, go and predict in that population, right? And also environment can sort of have a similar effect, right? If you drink coffee all day, perhaps there's a genetic variant that doesn't affect your disease risk because you drink coffee. There's an interaction, right? And there's actually such variants are known. There's, for example, Parkinson's and coffee. There's a variant apparently that decreases the risk of getting Parkinson's only if you drink coffee. So good reason for drinking coffee, actually. Good. I think a third thing that is sort of really important, and perhaps the most important, is try to convince the medical community that these predictions, that may seem sort of scary because they're using many factors, right? And you might not even understand the details of, biological details of each of these factors in these predictions, that they're actually clinically useful and valuable. And that, that sort of means that people like me and researchers that believe in these things need to advocate them and continue to do so, right, for some time. Right, and believing in polygenic predictions may also be difficult given that they didn't really sort of start off very well in human genetics. So in 2010 there was a big splashy paper in Science that claimed that they could predict whether you would live to be 100 with 77% accuracy based on a set of genetic variants, right? And it published in the New York Times and got some headlines, right? A year later, it was retracted. So apparently the 150 genetic variants that they found to be correlated with longevity, they had accidentally added the training data sort of in through the back door in the validation data, and they had some complicated validation scenario, and somehow they overfit the model. And when you cleaned it up and did a sort of a split up the data more carefully, the entire prediction just disappeared. So yeah, polygenic models, you have to be careful, right? You have to be careful about the validation as well. But the nice thing about polygenic model is that given enough data, the accuracy of these models, which depend on heritability, will be something like this, right, as a function of the sample size. So if you have a lot of training data, even for a highly, highly complex trait, you're going to be able to predict the accuracy of that trait to the limit of the heritability, if you just have enough data. So that's a nice thing about these polygenic predictions, and that's sort of regardless of the actual genetic architecture of the trait, even if it's just if it is a single gene that is causing the trait, it's sort of stupid to do a polygenic prediction perhaps, but it will still have the accuracy of that prediction will still become really good as long as you have enough data. And polygenic prediction really make a lot of sense for a lot of these common and, well, polygenic diseases, of course, right? And these are diseases like cardiovascular diseases, diabetes, breast cancer, many other heritable cancers, and many other diseases, right? And I think these three sort of categories of diseases, right, account for like 2/3 of death in the U.S. And at least the first two of them, preventive care can probably change the lives of a lot of people. So I think there's potential of polygenic predictions in medicine is tremendous actually. And already today for cardiovascular diseases, they already sort of use this risk score, this Framingham risk score that you might have heard of, and many other risk scores to try to predict disease risk of individuals. They're just not including the sort of polygenic component in a way that a breeder would, right? But I think sort of the community is sort of finally coming around to the same conclusion. What is going on here? Anyways, this should not have this fun, but anyway. Right. So this was a review published earlier this year in Nature Reviews Genetics. And here they just point out that these risk scores allow you to sort of, they argue that polygenic risk scores are useful, but they also point out that, you end up with these risk score distributions, that allows you to sort of categorize people into high risk and medium risk and low risk, right? And that's the aim, right? You want to identify the individuals for which preventive and this is a paper that is now on bioRxiv that I think is a really nice example of that is sort of convincing, at least for me, that polygenic predictions really make a lot of sense in predicting risk of coronary artery disease, I believe, or cardiovascular disease, right? So here, what they're doing is that they're comparing various types of risk scores. This is family history only. This is the Framingham risk score, which I believe is quite commonly used in clinics. Cardiologists use that. And what that does is it measures, various things in the blood, the lipids and triglycerides, and also they ask you if anybody of close relatives has had a heart attack, and that's a variable in the model. Then it's just some regression model that was sort of fitted on the Framingham cohort, and then they calculate the risk based on that, right? And this is the genetic risk score, which is, I guess, the red thing here, which is a polygenic risk score, right? And as you can see, if you validate in the Finnish cohort, this is a large Finnish cohort, you see that the polygenic risk score is actually doing better, and this is the difference in the accuracy onto the area under the curve, right? When compared to not adding it to the model, right? So basically, the higher this number, the more accuracy you gain by considering this as a cohort, right? So here the genetic risk score is doing better than just the Framingham risk score, and this is another risk score that is similar to sort of Framingham risk score. If you go into the Framingham study sample, there you sort of can see that the Framingham risk score is doing very well, which you sort of expect, which is trained on a Framingham sample, but the genetic risk score is doing pretty well and combined, like this is the combination. But what's interesting is if you combine both, well, you do better than both, right? The blue thing here is doing even better than just the genetic polygenic component and the Framingham risk score. And I think this is, one of these papers that may help convincing clinicians to think about these and using these types of risk scores in the clinic. And I think that is definitely going to happen eventually. Right. This is a similar paper that just came out a few months ago, I guess, or weeks ago, I think, in July. It sort of looks at type 2 diabetes. Here the prediction is also, here you also see individuals with the highest polygenic scores being the individuals with sort of the highest probability of being type 2 diabetes, getting type 2 diabetes, stratified by the BMI, right? But basically, if you're in the high risk category, you have substantially increased risk to get type 2 diabetes. And of course, as sample sizes, as the training samples get larger, right, the expectation is that you'll be better, you'll do better at, predicting these high risk individuals. So I think this is just sort of the beginning, the same thing here, and eventually this red dot here will start to move up and dominate everything. Or at least be very important, right? So I think these are sort of examples of, that this actually works in clinical settings. You can also try to predict height and BMI, and this is the UK Biobank data. This is a paper by Albert Tenesa, which I think is sort of has doubled in animal breeding or something like that. Basically, what they're doing is a huge G-blob. On the UK Biobank data, this is I think 120,000 individuals. They did a huge G-blob on that, and these are the correlations, accuracies, correlation between the predicted and the reported height, and correlation between the predicted. But yes, again, these polygenic predictions are getting pretty accurate predictions. This prediction is, for example, I think 40 or 30% more accurate than what they reported in a giant analysis that used 250,000 individuals. And here they only used 120,000 individuals. Right? And the giant paper, they did something else, but here the G-blob actually does perform quite well. So okay, so now I've talked a while about these things and these polygenic scores, but of course I need to talk about what I've been actually working on, since I'm also an academic. I need to sort of advertise my own stuff, right? So what I've been interested in is trying to improving these polygenic risk scores. And there are sort of two types of data that you typically have when you are doing prediction, in human genetics at least. You can either have access to all your genotypes, right, and you can just theoretically just do a G-blob, just like these guys did, right? Basically use this method that you use in animal breeding or to do a prediction. But often you don't have that type of data. You're only allowed to sort of look at the summary statistics, right? Basically, the people conduct these large GWAS meta-analysis where they combine GWAS results from a lot of cohorts and meta-analyze them to get new GWAS studies that have lots of significant hits and so forth. And then they make those results public, but not the genetic data. But there's actually a lot of information in these GWAS results, right? Especially because they're basing them on, say, for the height, giant data, a giant Jiva study, in height, right? That's based on 250,000 individuals. So that's the accuracy with which you can estimate these effect estimates of each of these SNPs is actually pretty good, because the variance scale is something on the order of 1 over the sample size, right? So the variance on noise is going to be pretty small, but of course the effects are also really small, so it's pretty difficult nevertheless. But so anyways, in theory, what you can do instead, if you don't have the genotype available to you, is, well, you just take the GWAS effects that are sort of marginally estimated, right, and multiply them with the allele, right? So you basically sum over all the allelic effects for an individual to get a prediction. It's not the smartest thing to do, and I'll come back to that, but that is something that you can do, and people actually do that, right? And you can actually show that if you do that, the expectation is, as long as you use the two causal ones, but a bit of a caveat, but you can show that the accuracy of that prediction will actually achieve the heritability threshold given enough sample size, regardless of the genetic architecture of the trait. And to show it sort of theoretically here. And that was sort of shown again by an animal breeder, I guess. Right, so what I was interested in was trying to understand what would happen if you threw LD into the equations, Linkwood's disequilibrium, right? So if you have unlinked SNPs, and P is the fraction of causal markers in the genome, so this is P equals 1. You can't really see it here. I don't know what's going on with the graphics, but this should be P equals 1, and P equals 0.1. So here, all of the markers in the genome are causal. So these are simulations, right? And 10% of the markers are causal, 1% and 0.1%, right? And the heritability is of the trait is here. So if you sort of optimize on a threshold, basically so that you only include the top SNPs and optimize on the threshold where you stop including SNPs, these are sort of the prediction accuracies that you can get. So if you have a less complex trait, only 0.1 markers are causal, it's sort of, you can predict that with decent accuracy using, not too many samples as training data, right? But as it becomes more complex, you need more samples, right? Makes total sense. If you throw LD into the equation, it looks a little bit different, and they sort of taper off way below reaching the sort of maximal potential, which is the heritability, right? And that's a bit of a problem. And the reason for that is that, well, because these GWAS estimates are marginally estimated, right? The reported GWAS estimates are marginally estimated, and you don't get the genetic data, so you don't really know the correlation between the estimates, right? So what happens is that they are sort of capturing the same things, and you're sort of including, basically getting slightly biased effects everywhere, right? And the more causal markers, the more of these biased effects are. Because if there are not that many markers that are causal, they're not going to be biasing each other very much, because they're usually not an LD, the causal markers, right? So I was interested in trying to sort of solving this and coming up with a better prediction, and well, of course, if you want to do a prediction, you can just open your statistics book and it tells you like what you want is the best unbiased prediction, right? That's really what you want, because that sort of assumes that. So basically, if God tells you like what is the genetic, tells you the genetic architecture of the trait. For example, that most of the genetic variants have no effect on the trait, but a few genetic variants have effect on the trait, and those effects are sampled from a Gaussian distribution, right? If I know that a priori, then I can try to calculate what's called the best bias prediction that is then guaranteed to be the optimal prediction. Obviously, that sort of makes this tremendous assumption that the actual genetic architecture follows this, but if it does follow this, then that's the best prediction. Right, so and you can write it up like this, and then I make some assumption. Well, first key assumption. Well, we're going to assume additivity, right? And you can move it on this side. All the contributions are additive. There's no epistasis in the genome, right? And then you can sort of try to estimate these things. And the way I did that was using some Markov chain Monte Carlo, the Gibbs sampler, and wrote this program LTPRET that then sort of approximates this. So, and sort of a key component here is this D here. I'm not going to go into the detail, but that is the LT matrix. And because of course you can't really account for the LT without knowing the LT. And the way we get the LT matrix, we don't obviously have the LT matrix from the data on which the original GWAS data was generated from, but we can use a reference panel. So make the assumption that the LT and the data on which the GWAS study was actually conducted on is going to be similar to LT of some reference panel. So we take a bunch of Europeans, because most of the GWAS studies are currently being done in sort of individuals of European ancestry, and then you sort of add that to the model. That was basically what we did. And of course, if you have a GWAS study that's done in African Americans, you would use African Americans as reference sample or something like that. But most of the large-scale GWAS studies are still based on individuals of European ancestry. Right. So we had this program, and of course then the next thing to do is to verify that it actually works and test it and compare it to other methods. And this is sort of the other approach that people have been using that sort of don't have the chance of actually seeing the genetic data and are sort of restricted to only working with summary statistics. And that method is really simple. It's just applying a p-value threshold that you optimize on and LT pruning, right? The LT prune set of SNPs, you sort of throw SNPs that are the prune SNPs that are in LT, retaining the one that is sort of more strongly associated with the trait of interest, right? So you LT prune and apply a p-value threshold, and these are the sort of prediction accuracies that you get. And this is a WTCC data set, 5,000 individuals or 4,500 individuals, roughly I think 3,000 cases and 1,500 cases and 3,000 controls for each of these diseases, and these are the accuracies. And I was very happy to see that the LTPRET was sort of doing better in some cases or most cases, but not all cases. But in most sort of cases, it was actually doing better than the rest. Then we also applied it to a couple of large GeoSummer statistics, but even though there are hundreds of them actually publicly available these days, it's difficult to find an independent data set that wasn't included in the original GWAS analysis, right? Because of course, if you have a height data set, you participate in the large GWAS height study, right? And become a co-author, right? So it was difficult to find a height data that was independent, that actually hadn't been, didn't take part in the large GWAS meta-analysis. But we're able to do that by collaborating with some people that had access to some data. And we also found that LTPRET was doing better than this P plus T. And there's sort of two versions of the LTPRET. And this LTPRET inferior, I should point that out, is really sort of analogous to a plot. It assumes that all of the markers in genomes are causal, and the sort of LTPRET is this Gibbs sampler that assumes that a fraction of the markers are causal, right? You could think about some of the Bayesian alphabet methods, right? There's an analogy to that, right? And the nice thing about the LTPRET inf is that you can solve it analytically. It's just an equation, right? Here you need to run a complicated MC that may or may not converge, right? So Fourier trait. Fortunately, it worked for all of these, and these were the predictions, and we see sort of nice improvements in accuracy for almost all of them. So we were happy and published this in the American Journal of Human Genetics last year, and we also ran this on height and BMI later in some Danish data, and this is just got accepted in genetics, and here this is sort of cheating by stratifying, plotting both the females and the males in the same plot. So actually, it turns out if you look at height, like 50% of the total variance in the population is explained by sex, right? But the variance explained after the accuracy of the prediction correlation after adding sex into the model and also principal components was around 25%. The correlation was close to 50% actually. The R squared was around 25%, which actually matches what they found in the UK Biobank data study. But of course we're using a much larger data set, but then again the training data is perhaps not as homogeneous as the UK Biobank data, which are all British samples, and of course we lose some power by having to restrict to achieve a summary statistics as training data. This is the BMI prediction, and there was around I think 30%, 0.3 correlation. This is probably also similar to what they had. Right. Anyways, a lot of people to thank. In particular, a lot of this work, or most of this work, was done while I was a postdoc with Alkes in his lab at Harvard. But there are also a lot of people that contributed data and some good advice like Podu and also Nick Patterson and many others. Right. And funding came from the Danish Council of Independent Research. So another project that I've sort of recently been working on, sort of a fun project since I moved to Denmark, is the Honest Gene Project. And this is sort of a, because I was really interested in trying to understand how you go from the genome to actual prediction, the entire sort of pipeline, right? If you were to actually, do genetic prediction in humans, how would the pipeline actually work? And what you need to do is you need to do imputation, obviously, and then or at least sort of to make things efficient. And then, ancestry is also important, because if you are, for example, African American, then it's very likely that the accuracy of these predictions that are based on training data from European ancestry individuals, right, is not going to be as good, right? So that's actually important. And then, seems like nobody has really implemented these polygenic predictions for humans, really. I guess there was these two recently published papers, but at the time nobody had really done that. So I was sort of interested in trying that. And we implemented all these things in a very efficient algorithm. And we supported these genotype formats that you might have heard of, or perhaps you have your genotypes in these formats. And basically, using our framework, we can go from genotype to prediction in about 2 minutes, which we are very proud of, because I don't think anybody else can do that at the moment. So yeah, I'm just going to see if this actually works, show you, sort of guide you through this. I was testing it earlier, so I'll delete this. This is actually a cookie. The genome is actually never stored on the server, so we just delete it after running analysis, right? So I'm just, so it works like this. You consent something, and basically, this is sort of this consent form is is strongly sort of influenced by the DNA length consent form at the moment. But this is of course beta, so this is not really the final version. But then you upload your genome, or if you have a 23andMe account, you can also just do it through that, and then take some time to send the data to the server. And after a couple of seconds, depending on the internet connection, then it should start.
How many is this?
So it's the 23andMe data. So there are 4 versions of the 23andMe data. This I think is the version 4, which is 500,000 SNPs. I guess I could continue a little bit. Right. Yes, is it legal? That's a good question. So then you impute it, starts imputing. You talk about whether it's legal. So that's a serious concern, especially since up until recently, 23andMe wasn't really allowed to do any health-related predictions. So that was a concern. But it turns out that we believe, in Europe at least, that this is legal because we're not doing any disease prediction, right? But the EU Parliament is actually currently discussing whether to just ban direct-to-consumer genetic testing altogether. But it's, why is this interesting? It's open source, so basically anybody can look at the source code and download it. It's easy to use. We try to make a good documentation so it basically can be set up anywhere for various different purposes, right? One instance could be that you could repurpose this whole framework for genetic testing in clinical settings. It can also be used to recruit individuals for studies, similar to what 23andMe is doing, and give individuals incentive to participate in studies, and perhaps also provide a more informed consent, because individuals are actually allowed, they can look at their genetic data and perhaps learn something about themselves and how these, what you can do with this genetic data. Can I see it? It's actually right. So now it's imputed, and the ancestor is calculated. The visualization is a bit lacking. And here we just did some PCA, and unfortunately I haven't gotten my 23andMe genome yet. It's currently sitting at home. I haven't sent it yet, but I need to mail it in. But this is a colleague of mine who's Finnish, and I don't know if you realize that this is 1,000 genomes. So these are African and African Americans in the 1,000 genomes. These are East Asians, South Asians, and these are Hispanics, basically. The magenta, and so basically here the PC is differentiated between basically 3 populations: African, East Asian, and Europeans, and not doing a great job of therefore distinguishing, Hispanics or Latinos. But this is the Finnish cluster, so he falls very nicely in the Finnish cluster. This Finnish individual, and then you can do a prediction. Currently, we just implement height and BMI, but actually, then you get some prediction, and this is your genetic prediction, and the validation data here, which is important, is Danish samples, the same Danish samples that I used before, so if you're not Danish, we probably can't guarantee your validation. But if you are a Danish high school student, I think we can accurately say that it's probably 10 to 20% variance explained. But I just thought this was a fun project and thought it would be fun. I don't really know how to publish this yet, but thinking about it, various things that you can do with this. I think one thing where you could use this in education and also to try to get students interested in studying genetics because, well, you can look at your own genotype and actively sort of write code to analyze your genotype and do it in a visualization framework. Right, I think about this also as a complementary analysis to 23andMe. Hopefully we can, tell, well, we can certainly predict height better than they can because they only look at 3 SNPs that explain, I think, if you have all of the variants, they explain about a centimeter or half an inch. Right, so I think we can do height prediction probably better than they can. And we implement a polygenic prediction, which is, not what they do. They only look at the individual SNPs. We plan to implement the predictions for more fun traits. We're working on getting data for that. Currently, we're actively avoiding disease risk prediction because the medical community in Denmark tells us that we'll probably be labeled crazy if we do that, and I don't want to be crazy. Right. So there are a bunch of people also to acknowledge on this project. In particular, actually, the guy who's actually particularly is not listed here is Emmet Seren. He's so obviously supposed to be the first person here, but I forgot to write his name. And a bunch of other people. And this was also done while on this funding from the Danish Council for Independent Research. Thank you.
So we ran a little bit over time, so maybe time for 1 or 2 questions. We perhaps can have more questions at the end during lunchtime if anyone's interested. Well, save them for lunch then.
All right. Thank you.
Thank you.
So in the meanwhile, I'm going to introduce Dr. Paul Van Reyden. So Dr. Paul Van Reyden is a research geneticist in the USDA RS. And let me find your presentation. I don't know if it's already open. Right there. He also has been working in methodologies to do genomic prediction, actually, and I'm probably the one that he worked and he did, the greatest contribution, I think, for all the communities, kind of the development of an easier way to do genomic prediction, basically through the GBLAB. A work that actually has more than 1,000 citations already, even when it was, just published in 2008. So he was just telling me that he started working on that in 2007. Actually, it came from a large conference in animal breeding, I believe, or genetics from Brazil, because they were saying that the chip was coming, basically, and the 50K chip was coming for animals, and they were not preparing how to use it. And basically, that's when he started working on that method that he delivered, a very good methodology that I think is one of the most useful methodologies in genomic prediction. It's a pleasure to have you here.
Well, it's a pleasure to be here, Patricia. Thank you for the invitation to come and the introduction. This talk will follow very well along with the last one and the ones before. Somewhat different species, but a lot of the DNA-based methods are becoming very, very similar tools being used by everyone. So genomic prediction, it sounds like a wonderful new tool, but you actually have to have some input data to be able to do it, and we were fortunate in dairy cattle to have a lot of preparation time so that when the new tools became available, we could take advantage of it right away. But to start out similarly, where did these ideas come from? Well, a lot of genetic theory was actually developed in the agricultural sector. Sorry, that's the next slide. This one is just to get you a flow of where the talk is going to go. So why did genomic selection get started so fast in dairy cattle? What was our industry like before genomics came along? Now, what has changed? The methods we use for prediction, and some of the trends that have took hold in the last 8 years or so. So some of the theory, obviously Gregor Mendel was working in agriculture at the time when he discovered the single gene inheritance. The multiple gene or polygenic version of inheritance, the data in the 1918 paper from Fisher was actually human traits, such as you just talked about. And the pedigree relationship matrix, which has been a key feature of what we use. The example for that was a cattle relationship matrix from Sewell Wright, working at the same lab, basically where I work now. The mixed linear models were developed by Henderson over many years, mainly working with cattle, and that includes using the inverse of the pedigree relationship matrix. The classic Mewison paper and a few other ones on how to do genomic prediction, those were mainly using animal data. And the genomic relationship matrix, I was trying to get ready for dairy cattle data, and I'll give you some, well, you've seen many results already today on that. So, but not only the theory, but partly the reason we developed these tools is because of the application that we have to do. So, for example, 1 year ago, we believe we were the first ones to reach 1 million genotyped individuals on our joint database held by USDA and the Council on Dairy Cattle Breeding. At that time, 23andMe had 900,000 genotypes. Ancestry.com had 800,000. So, but things change very quickly in this business. And so now, all of a sudden, a year later, we've been, left behind. So, Ancestry.com now has on their website more than 2 million. I believe that's the largest one. We're up to 1.4 million at this time with cattle. 23andMe had reported, at least 55 countries that they get DNA from. I think probably similar for Ancestry.com, and we're up to more than 50 countries receiving cattle genotypes. The cost, it is possible that they're starting to do these predictions. I think the $99 cost is just to get the information about where did your DNA, where did your ancestries come from. The 23andMe, for the higher cost, also does some predicted traits. We have a range of prices depending on the density of the chip that they order, and that includes both predictions for like 30 different traits, genetic tests for lethal conditions, and also which breed did the DNA come from. So the one big difference is on DNA generations. There's going to be lots and lots of ancestries genotyped in our database, and very few in the human databases. And as a result, we can get high accuracy of predictions, and that's why the growth was so fast on the customer use. So why has genetics and genomics been so important in dairy cattle? Well, one thing is that the bulls do not directly express the traits. Milk production is a trait of females. The bulls are reproductively the more valuable, but we have no information other than doing statistical analysis to try to to use the genetics to determine that. And the cows themselves are worth about $2,000. So if it costs, $40, $50 to do a genetic test, that's fairly reasonable. And also because the owners collect a lot of data to manage these cows. So we've had that for 100 years, data flowing in because the farmers collect it automatically. And we have also pedigrees for about half of the U.S. cows, so 9 million cows. We get new pedigrees every year. It's like more than 70 million in our database. And this has been going on for more than a century of national data collection, which is not the case in most other species. So the system that was used from about 1950 until genomics came along in the last decade was you would have a bull like this. They would get a sample of daughters, and based on the analysis of those first daughters, we would decide, do we want to use the bull some more? And using the bull some more meant for this one, he has more than 100,000 daughters that have milk production records, 60,000 in the U.S. and 40,000 in 25 other countries. And that data is all collected, through these national databases. And this bull sold more than 1 million units of semen, probably worth more than $30 million. And of course, he was one of the very best ones out of 10,000 that were tested. So once we find the best one, the farmers all want to have daughters of that bull. And so why would they want to use that bull so much? Well, the traits that our predictions are that the daughters would give 6% more milk than if you would use an average bull. 17, we don't predict that they'll live to be 100, but we do predict that they'll be 17% longer in the herd than the average bull, 10% more fertile, 50% fewer difficult births. So we have many, 30 traits like this. And if you add up the value of all those 30 traits, you get $700 more profit than the average cow. And that's actually the way we express our overall index. And so if you have 100,000 daughters, each making $700 more profit, you get 70 million. So you can see why they paid 30 million if they're going to get 70 million profit back. That's just for the first generation. The traits that we've included back in 1971, just milk and fat production, then added protein, longevity, mastitis resistance, some conformation traits, calving traits, fertility traits. This one we plan to add later this year, livability, is does the cow actually die? Not just is she sold for beef, but does she die on the farm? So we have several more traits that we'd like to get started, and genomics makes those fairly easy to predict. Another big reason why we can get started quickly with prediction is you have to have DNA on the same individuals that you have the phenotypes on. Makes the analysis easier. And in this case, we had central DNA storage for basically all the progeny tested bulls back to 1992. And these were all the AI, the companies that owned the bulls were sending DNA to USDA Beltsville. And these are basically all the major companies in US and Canada, but they're also global companies. They do business around the world. So we had the DNA storage, those bulls probably sire about 80% of the U.S. cows, and $150 million per year of semen exported to other populations. Then in a lot of the other countries, they are using their own bulls locally, but those bulls are generally sired by a U.S. bull. So they're only, 1 or 2 generations removed, the cows in the other countries from ours. And this is a historical from the 80s, 90s, 2000s. This is the percentage of the foreign bulls that had U.S. sires, and now that when we started genomics, it actually went back up again. So the genetic progress here is really driving the progress in a lot of other places. So this is just the progress of how we got started in markers. We spent 13 years collecting 367 markers on 1,000 animals, and never really made any use of that practically because it just was not very accurate. And then we got a SNP chip first in 2007. First publication, we had up to like 16,000 animals. Very soon after that, we went to a high-density chip and 10 times as many animals. This is what we routinely use now in terms of number of markers. So we've added some of the best SNPs here. We've added to the lower density chip and made a bigger analysis with more than 1 million animals. And last year we got 39 million SNPs on 1,500 bulls from the global 1,000 Bull Genomes Project. A lot of these are all international efforts. So one thing that did take us by surprise is the rapid variation in input data. So we went for the first 3 years with the 50K chip, and it was slightly revised. But then high density chips, very low density chip, and gradually we have gotten more and more companies providing more and more different SNP sets. And we have to put all this data together with imputation. Makes a very big, complicated analysis and do all the quality control. And in the last month, we added 2 more chips. So it doesn't seem to be any. That's even before we started putting the sequence data into the mix. So what is the idea? Well, we used to, a long time ago, just look at the animal's own traits or use pedigrees and phenotypes. And since 2008, we've been looking directly at the DNA, either to predict for a calf what she will be like as a cow, or more usually, what will the next generation look like. And of course, there's been various improvements in the statistical methods and these are the official models used by USDA over the years, gradually improving, except this maize multi-trait across country evaluation. We get the information from all the foreign data through Sweden, through Interbull for about 30 different countries. So that's been, since 1995, we've had global data, basically. And the last thing here is that we are gradually replacing or adding these causal mutations instead of just markers to use what we believe are real QTLs. And to get ready for being able to fully take advantage of the genomic data, revised in 2014, completely rewrote the software. We had been gradually revising and revising old software. Well, finally we came up with an all-new model with much more flexible statistical methods and also parallel processing to speed the thing up. And we were hoping to be able to go to the single-step model where we combine the genotypic, phenotypic, and pedigree data all together. One problem there is with more than 1 million genotyped animals, the G-inverse gets somewhat difficult. So there are ways to get approximate G-inverse now. I'll talk about that in a little bit. This is our current multi-step system, which so we have some pre-processing steps, computing the inbreeding and heterosis for 70 million animals. Then the traditional EBVs, this is just the phenotypic and pedigree portion, and that is used as input to the genomic system. And imputation is one of our biggest or longest steps. Calculating the SNP effects for all the traits, compare the G matrix to the A matrix, and so all of this together is less than a week using no more than 30 processors. So that's fairly doable, and but doing single step is more work, especially if you're trying to use a G inverse. This is using SNP. So we actually use the Bayes A. I'll show a picture of that in a little bit. And assume when we say polygenic, that means the part that's not accounted for by the 60,000 marker effects. So 10% still remains on the pedigree portion that's not accounted for by any of the SNPs. And these are some equations still from the 2008 paper. I think probably they're fairly well known. And so maybe we'll just skip those. But everyone refers to that as a GBLUP paper, but I don't actually use GBLUP. We've always used this Bayes A. So instead of just using a straight line regression with equal weight on all SNPs, it regresses those with small effects, regresses them closer towards 0, and those with bigger effects, it lets them have more of the variance. So that's, it's not a big advantage, but it was, you know, a 1 or 2% advantage, so we've decided to use that. So on to the single step. Obviously now the assumptions in our previous equations, the pedigree and phenotype only, are violated because the animals that are getting the data are pre-selected based on their genetic effects. So the expected value of Mendelian sampling is not 0, and so many studies have shown that you can improve the accuracy and reduce the bias. But how do you get the system to work when you have 1 million genotyped animals? That's been one of the key things we're working on and trying to figure out. So exactly what are the biases that can happen if we continue to use our multi-step model instead of the single-step model? Is that we're correcting the value, so these animals are mated together to produce the next generation, and it adjusts the merit of the either parent for
for the other, so you're isolating that animal, that parent's contribution. But if the selection was actually based on the GPTAs or GEBVs, then you're not fully accounting for the selection of the mates. You're not fully accounting for the genetic merit of the herd mates. That can underestimate the genetic trend. And also, if the only progeny, say, that get phenotypes are these highly selected ones, it's going to make the parent look way too good because, oh, look at all of this, you know, these progeny are so wonderful, but they're only the ones that were pre-selected. So several studies have derived equations to account for the genomic effects through the G inverse at the same time as the pedigree relationship matrix for all the animals by dividing the equations into the genotyped animals and the not genotyped animals, come up with this set of equations from Ignacio Aguilar and Ignacio Mistel and the group in Georgia. And others have, Christensen et al. Have also derived very similar equations. So what we were waiting on is, can we figure out a way to process 1 million or more animals? Ignacy and his group have figured out the math to do that using approximate G inverse. Yutaka Masuda has applied that algorithm, and in terms of computation, it is very reasonable, both in terms of memory, processors, time. It's affordable to do. But we noticed that it had less bias than our current system. So that was the good feature we were hoping for. But the accuracy of prediction started to drop as we got all of these young animals with only genotypes but no phenotypes were added in. So we're not sure exactly what's the cause of that. All right, so this is just of interest. I said we had lots of generations of genotypes. So this is a pedigree that came in of an embryo that was actually genotyped. This is the sire's pedigree. So the sire genotyped with 77,000 SNPs. The dam with 9K, 50K. You can see the parents, grandparents, great-grandparents, great-great-great-grandparents. I mean, it's just basically complete genotyping all the way through. These ones that say imputed, the cow herself was not genotyped, but she had so many progeny that were genotyped that we can reconstruct from that. And if you go back 8 more generations, almost all of the males in the pedigree are also genotyped, and with phenotypes. So this is the data we have to work with. That's the male side of the embryo's pedigree. This is the female side, looks the same. It's just, I did not imagine this 8 years ago that we would be at this point. So that we have all these males and females genotyped. Then we had a postdoc, Xuanyu Sun, that worked on can we provide genomic relationship matrix genomic relationships instead of pedigree relationships for the mating programs. And a lot of cows are assigned their mates based on inbreeding calculations to try to avoid inbreeding. So we have, right now we send out for nearly 1 million females and about 5,000 of the marketed males. The genomic relationships And that we've been doing since 2014 for people to use that in their mating programs. Another thing gets the farmers interested or more knowledgeable about what's happening. This is a chart he happened to be our highest net merit bull at the time this was made. And you can say, well, why is he such a good bull? Well, he had a great chromosome 1, very nice chromosome 28, very nice chromosome 10 and 6, but there's room for improvement here. Even the best bull, he's got you've got a below average chromosome 14, 16, 20, 27. So obviously we can make improvement if we find some animal to mix the genes in, which is outstanding for the chromosomes where he is poor. Since then we've phased the chromosomes, so we actually provide the maternal and the paternal individual chromosomes in the chart. I didn't happen to get that one, but those are online. And every farmer can type in the number of their animal and see the chart for their animal to see which chromosomes are good and bad. A lot of them do. They're just curious. So then if you think about it, if you take the best chromosome from Freddie, the best chromosome from somebody else, and you assemble them in a petri dish or however you would do it, what's the best animal we could make? And you come up with 7,000 or like 10 times, so we can project. If we assemble these chromosomes, the best ones, we could go 10 times further than we've already gone with the best animal now. So this, besides trying to get the best animal, the other question is how fast can you do it? Well, we don't have Petri dish method working yet, so we just have to keep making matings and trying to choose those with the best chromosomes. And this is another chart, but this one is across time. What were the age of the parents at the time their calves were born, so the generation interval. Now we used to wait, based on progeny testing, we would wait about 7 years before these bulls would have their sons born and be used, because we had to wait for the data to accumulate. And in the last few years now, more and more of the bulls are being used right away when they're yearlings, so that the calves are selected and used or chosen when the bull is only less than or around 2 years of age. And females have not come down as much, but this is one of the main gains of genomic selection, is to reduce the generation interval, and they've really done a good job of that. So this is more of a historic time trend in terms of what is the progress we've been making for this overall net merit, the dollar value of all the traits. And the genetic progress was fairly constant over the years, and you can see that when genomics came along, the trend in the bulls has gone up very steeply, and usually the trend in the cow population lags a generation or two behind, but we expect it to be going up, almost at that rate within another generation or two. To look a little bit more about what our predictions, this is the data that the farmers are waiting for. We just released our 3 times a year major, major update this month. If we look back to 2013, these are the young bulls. So we predicted them. At the time, they had no daughters. They were just young calves. And now they have 1,000 daughters with phenotypes. So these are in order that we predicted them at the time. This bull Super Sire turned out to be much better than we predicted. Even the second best one was much better than we predicted. Most of them have been fairly consistent with the predictions, but so that gives the farmers confidence that yes, they should use these young bulls, and they have used them to such an extent that now the young bulls are getting thousands of daughters right away, and many of them are used as parents of the young bulls in the next generation. They still can use wait until the bulls are a little older and use the proven bulls based on daughter records, but then on average those bulls are just not as good because they're a generation behind by that time. And some of these still do end up with, many daughters and many sons, but increasingly the usage is very early in life, not waiting until the bull actually has daughters with phenotypes. So this is just a comparison of the different breeds. Holstein are the biggest breed in terms of reference bulls. You can see that the reliability, which is the squared correlation or the estimated squared correlation between the EBV and the true breeding value, that's getting up there to where the young bulls are becoming almost as accurate as the proven bulls, whereas in the smaller breeds, the genomic prediction is still closer to the original parent average. There's just not that much additional information in those. So the smaller breeds do have a little bit of a struggle to keep pace with the bigger populations. We've tried to get as much data as we can by exchanging genotypes internationally. From the beginning, we've exchanged all breeds, both sexes, with Canada. They have a copy of our database. We work with the same data as far as genotypes. And then we also added all the bulls are exchanged with UK and Italy. That's been going on for 5 years already. In the brown Swiss breed, that's these cows. For that breed, we've exchanged all the bulls via interbull for a few years. Exchanged Jerseys with Denmark, Guernseys with the UK, and we've recently added just this year all the proven bulls from Japan and Switzerland, and the young bulls from Germany. So this is data is flowing in many directions. So, all right. The other thing surprising is the customers. This shows the countries that have sent in genotypes from their cattle to get tested in our system. Very easy to do. And there's just a small calculation fee. In terms of where they actually, how many from where, North America is still obviously the main source. Western Europe, more than 100,000 from basically every country. Eastern Europe, Australia, New Zealand, about 10,000. Most of the countries in Latin America have sent thousands of genotypes in. Several countries in Asia are now sending in genotypes, and a few now from Africa too. And so obviously this is exponential growth. Our maximum upward potential with cattle is certainly not as high as the human databases can go. We run out of cattle before they run out of humans. And in terms of how the DNA physically is collected, most calves are given an ear tag, hopefully right when they're born, and then they can collect DNA right at the same time. The sources of DNA sent in, it used to be hair roots were the most popular, but recently it's more ear tissue. Those are the two main sources, but so the labs will accept many different tissue sources. This is an example from one farm. They went out to collect the ear tissue from their calves. This is a big job of DNA collection. Nobody has given me an exact count of how many were in there, but you usually don't have to measure DNA by the pound, but it was sitting on a FedEx scales there. So. Okay, last topic for me is sequence. Until a year ago, I had never seen any sequence data, so I tried to study up and see what should we do with it. And there's two main steps. The first step is alignment, where you take chopped up DNA and try to figure out where does it map to on the reference maps. And that's usually from paired end data, where you map this end and you map that end and make sure that it makes sense along the chromosome. And then the second main step is the variant calling, where you try to figure out, is there a difference from this piece of DNA that I just aligned to the reference map, and those are the variants. And they could be either ones that somebody already reported, or some that's completely new to this individual. And the programs that are available generally always do alignment first, and then a later step is to do the variant calling. And so I wondered, is there an advantage to doing the two together at the same time by knowing not only the reference map, but all the known differences from the reference map? Hold on to that information in memory so that you can match more than, you have the opportunity to match not just the reference genome, but all the other variations on the theme. So we tested BWA and GATK, and I was surprised that the main GATK paper says this strategy guarantees that many reads spanning indels will be misaligned. So it seemed like, oh, well, maybe that's a hint that doing them together would be better. And so this is the reference genome. Oops, sorry. The reference genome for cattle is this inbred Hereford cow. And so you align, every piece of DNA is aligned to see how well does it fit and where does it fit in her genome. But my new program, FindMap, can also know all known differences within and among breeds to say, well, if it doesn't fit her genome, maybe it fits one of the other animals that's been genotyped. And so the error rate is reduced then by using the known SNP and indels to separate those from the machine read errors during the alignment process. And then it's still mapped back to the same common reference map. In terms of timing and accuracy then, compared to BWA, it was about 60 times faster with one processor, and it shares memory among processors. So if you use 10 processors, it's about 30 times faster. The memory use to hold on to all this extra information, the memory was larger, but it also can share that memory, so the total was not really any different. And the variant calling was also much faster. The correctly placed segments was higher, as I had hoped for, and more of the paired ends were correctly placed. And so this looked like a optimistic, new tool to try. I don't have all the fancy features, but I did submit a paper on this just this month. And then as far as the use of sequence data, fortunately, many researchers in the whole world have joined their data in this 1,000-bull genomes, which is managed in Australia. And we got the data for 440 Holsteins, imputed 27,000 reference bulls, and just used basically the candidate SNPs within the near genes. Estimated where the largest ones were, added those to our routinely used, and found a possible gain of 2.7% reliability, which that seems like it's going to pay for the extra work right there. And we've put some of these on several of the low density chips, so some of those should be coming out in the next few months. To conclude then, examining DNA seems like an excellent idea. Selecting young bulls gives much faster progress than our previous methods of progeny tested bulls. We're certainly adding more animals and more traits all the time, and that improves the accuracy and the value of the whole system. And our database is similar in size to the largest human databases, but we feel like it's much more valuable in terms of customer satisfaction. So, are there questions? Oh wait, I got acknowledgement of my own lab. There's many people in my own lab that contributed to this talk, and this very last slide on the 1,000-bull genomes is the group in Australia, and all the contributors of data to that. Thanks.
Questions?
Well, with platforms like the HiSeq 10, do you see this rapid change in platform genotyping that you've used before now rapidly potentially changing to a whole genome sequencing approach?
Well, the fact that we went to these reduced SNP chips, they want the really low cost. That's been the big growth area. So sequencing for research is certainly very attractive. We expect that that's going to continue to increase, but commercially, not sure that the price is quite there yet.
What's the lowest price right now for the chip, for the low density chip?
There's some the full service can be less than $40. It could be, maybe a couple of years we'll be there with the sequencing, or at least with low coverage sequencing.
Very impressive talk, thank you. So one question that I have is, how do you handle, with so many data points and so many generations, how do you handle the all the mutations that show up that perhaps are important, both from a phenotypic perspective and also from a genotypic perspective that might not be included in your genotyping panel?
Yeah, that's we've had a little bit of experience with from these low density chip panels, we don't really discover a lot of the new mutations, but but we've had a few cases where researchers have identified within, between generations, because we have all this pedigree data, that between this generation and this generation, a new mutation, which was actually a lethal mutation, happened. And the only way we found to track those is to continue to use the pedigree, unless you're going to do sequencing all the time. Either you can put that new mutation onto one of the chips, but then you still don't have it on all the other animals, the other million animals that you've already tested. So our imputation algorithms use the pedigree so that we know the mutation is in this side of the family, but it's not in these other families over here. And that, for those new mutations, I think the pedigree becomes much more important.
All right. So we typically hear that G/E is low in animals. Is that, looking at your data, is that the case even for like extreme or cross-country environments?
Yeah, the interval system does correlations across all of the 30 countries. And for a lot of the traits, the genetic correlations are above 90. So that would indicate not a lot of G/E. I just presented yesterday to the animal science department where we've done G/E within the U.S. in terms of hotter climates and cooler climates, and got some statistically significant re-ranking, but still the correlations were pretty high. So in dairy cattle, they're not as subject to the environmental stresses that a lot of the other species are. They're managed in a more confined, controlled environment. That may reduce some of the interactions.
So I was thinking about the single step club, right, where you sort of combine the genetic and the pedigree. So you said that 10%, some slides said 10% of the added, you get 10% more value by adding the pedigree or something like that?
10% of the variation wasn't accounted for.
Oh, wasn't accounted for,
yeah. So when we first started the genomic predictions, we assigned all the genetic variants to the SNPs.
Yes.
And that tended,
partitions like that.
it tended to over predict in terms of the regression. We were predicting the top bulls too high and the poor bulls too low. And one of the ways we got that corrected was to assume that the SNPs don't account for all the variants and put some of this 10% back on the pedigree relationship.
Right. Okay, so I'm curious, like, is that capturing genetic effects, do you think, or is it capturing environmental things? Because I mean, if you have closely, bulls, I don't know, in humans at least, closely related humans, they tend not to grow in similar conditions. But of course, with bulls, it might be completely different, right?
Yeah, we hope it's capturing additive genetic variants.
But is there a correlation between environment and relatedness?
It didn't really help the accuracy. It was really, and sometimes it would actually hurt the accuracy a little bit, but people did not want these predictions to be, telling them things that weren't believable. And so it was partly just to get the scale back into the same size as the standard predictions.
So you talked about the figuring out which cow to mate with which bull, basically to reduce the inbreeding of the calves, right? Does that also, and then you talked about designing the super cow that had all the best chromosomes. Do these two things sort of go together? I mean,
well, yeah, that's a good point. And John Cole did that study on the super cow, and so we wondered, is it better to go all the way to 2 copies of the best chromosome, or should you hold off and take 1 copy of the best chromosome and 1 copy of the second best chromosome to avoid inbreeding? And the math worked out to say, go all the way to homozygous for the best chromosome. That was, we're a long way from getting there at the moment, though.
So it's sort of related to the same question here. So you know what is the optimal animal, right? You know what is sort of the perfect genetic makeup. Could you now, based on the fact that you know all the pedigrees or a great deal of all the genotypic data, could you try to trace back what would be the perfect pedigree that would lead you towards that goal? Understand? Sort of what would be the pedigree that I would need that would be the most probable to give me that genetic makeup?
Yeah, so I mean, in terms of, I'm not sure if you're really being looking backwards or...
No, I mean you would have to
yeah.
go forward.
So looking forward, and the idea of how can we think of it as an experiment, how can we most quickly assemble the chromosomes that we need to make that perfect animal? That is a reasonable research goal that I don't think people have really done that much simulation with. But instead of
It's a simulation exercise.
yeah, it's a good simulation exercise, and probably it helps you think about where should the population go, and is it better selecting for a good chromosome, or should you hold out for the best chromosome? Because if you select for the good chromosome, you're just going to have to replace it with a better one later. So you increase its frequency and then may have to decrease it later.
So I saw you put a comment about dominance, and if somebody probably can study dominance with a big data set, it's you guys. So what can you tell us about non-additive effects? Where are you guys finding it?
Yeah, we did that at the same time as the mating program. So the inbreeding was one thing, and the individual, dominance, those are very easy to solve for because you just like SNP effects, you just have one effect in your model for each SNP. And we found some use in some of the traits. It was not as important as just the overall inbreeding. And I think we decided that if people really wanted it, we could make it routine, but we haven't gone to the point of making it routine because maybe at half of the traits didn't seem like there was really enough. We couldn't estimate a big enough variance component to justify the calculations, but there were maybe half the traits where there's at least enough dominance to make the calculations pay.
So in QTL studies, typically if you try to pyramid 2 favorable QTLs, you find a total effect that's less than the sum of the favorable individual effects of the QTLs, sort of synergistic epistasis or anti-synergistic epistasis on this. And so that occurred to me when you were discussing the super cow. I mean, presumably not all chromosomes. Well, I mean, I wonder if there's a way of studying this. Like if you took your data back to the arbitrary date 2003 and you estimated what were the best chromosomes from 2003, now 13 years later, whatever, we must have some animals that have where some of these good chromosomes have been assembled together.
Yeah.
Is the effect less than you would have predicted based on 2003 data or whatever? You see what I'm getting at?
Well, what I've always wanted to do is also fit the additive by additive. That would at least, I think those interactions could be important there. Those are mathematically more difficult because the number of interactions is, even if you have 60,000 SNPs, it's 60,000 squared potential terms if you do it that way. We also have used, Jeff O'Connell has done this, used you have the G matrix, which is the additive effect, and then you have maybe a D matrix, which is the dominance term. And you can put also additive by additive which is only the number of animals by number of animals, and solve it that way. That works fairly well, but our variance estimates don't seem like, when you try to put those things, they're like additive and additive by additive are too confounded, and we don't give very reasonable estimates. So I don't have a lot of good experience with that, but it certainly is worth trying again.
For a given trait, what's the average mirror sense heritability?
For our traits, like stature is one of the higher ones, close to 50. The milk yield is in the 20s. Some of the other traits are in the teens, but the fertility traits are around 3%. This new trait we're going to add this year, the livability, is only 1%. So those are the kind where before we said, oh, there's not enough genetic variation even to worry about. But genomics has really amplified the power so that now we can pick up signal where we would have given up before.
So I noticed that early on you had a table that showed how much weight is being put on each trait, and at this point there's actually negative, very slightly negative weight on milk yield. Is that right?
Yeah.
So does that mean that it's just not worth any? I mean, it's not worth it, or is it that all of the net merit is coming from other traits, and are cows continuing to produce more and more milk given that?
Yeah. So that's because most of the milk, other than in Florida where it goes mainly for drinking milk, most of the milk is going to cheese or ice cream or powder or something like that. So the solids, the protein amount and the butterfat amount, those are getting big positive emphasis. Then the milk itself is more measuring how much water is there along with the solids. So if you're sending your milk to a cheese plant, there's actually no point in the cow putting water in, hauling the water to the cheese plant, and having the cheese plant have to remove the water. So it's really it's the leftover portion after accounting for the solids.
Okay, well, thank you, Paul. This concludes the 2016 version of the workshop. I need to thank you, the speakers, for coming and for kindly agreeing to make the recording available in the future. So that will be available through the website. If you want to miss some of the talks, you want to watch it later, these will be available. We will organize another version of this workshop next year. If there are any suggestions of names or different changes, feel free to provide those. And again, we'd like to thank you, the sponsors, and all of you guys for coming here. I don't know if Patricia has
that would be all. So I mean, I just one more opportunity. I guess we have 5 more minutes to, if you guys have any other questions for any of the speakers before that you never thought about that before. But in reality, we have a very good workshop. I mean, we discuss about new methodologies and how to incorporate, biological information in the prediction models and how improve. How that can make a big impact, in a crop like cassava, for example. And what are the ethical and legal consequences of using these methodologies in human genetics? And obviously a very successful story on how genomic prediction is working in cattle breeding, and how not only a success story from the point of view that it's using is how the farmers are actually incorporating and integrate that into their programs and in their farms. I mean, I have met farmers actually talking about genomic prediction and everything, and which is amazing. And that's something probably that in all the other crops that we are working with is something that we're looking forward. So having this group of people here, great researchers and scientists come in and tell us these stories, I think is very valuable for not only the scientists here, but also for the graduate students to see what is the potential. So thanks all for coming.